Section 8 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. The Hindbrain, Part 1. The encephalon, or brain. General considerations and divisions. The brain is contained within the cranium and constitutes the upper, greatly expanded part of the central nervous system. In its early embryonic condition, it consists of three hollow vesicles termed the hindbrain, or rhombencephalon, the midbrain, or mesencephalon, and the forebrain, or prosencephalon, and the parts derived from each of these can be recognized in the adult. Thus, in the process of development, the wall of the hindbrain undergoes modification to form the medulla oblongata, the pons, and cerebellum, while its cavity is expanded to form the fourth ventricle. The midbrain forms only a small part of the adult brain. Its cavity becomes the cerebral aqueduct of Sylvius, which serves as a tubular communication between the third and fourth ventricles, while its walls are thickened to form the corpora quadrigemina and cerebral peduncles. The forebrain undergoes great modifications. Its anterior part, or telencephalon, expands laterally in the form of two hollow vesicles, the cavities of which become the lateral ventricles, while the surrounding walls form the cerebral hemispheres and their commissures. The cavity of the posterior part, or diencephalon, forms the greater part of the third ventricle, and from its walls are developed most of the structures which bound that cavity. The hindbrain, or rhombencephalon. The hindbrain, or rhombencephalon, occupies the posterior fossa of the cranial cavity and lies below a fold of dura mater than tentorium cerebelli. It consists of A, the myelencephalon, comprising the medulla oblongata and the lower part of the fourth ventricle, B, the metencephalon, consisting of the pons, cerebellum, and the intermediate part of the fourth ventricle, and C, the isthmus rhombencephali, a constricted portion immediately adjoining the midbrain, and including the superior peduncles of the cerebellum, the anterior medullary velum, and the upper part of the fourth ventricle. The medulla oblongata, spinal bulb. The medulla oblongata extends from the lower margin of the pons to a plane passing transversely below the pyramidal decussation and above the first pair of cervical nerves. This plane corresponds with the upper border of the atlas behind and the middle of the odontoid process of the axis in front. At this level, the medulla oblongata is continuous with the medulla spinalis. Its anterior surface is separated from the basilar part of the occipital bone and the upper part of the odontoid process by the membranes of the brain and the occipital axial ligaments. Its posterior surface is received into the fossa between the hemispheres of the cerebellum, and the upper portion of it forms the lower part of the floor of the fourth ventricle. The medulla oblongata is pyramidal in shape, its broad extremity being directed upwards towards the pons, while its narrow lower end is continuous with the medulla spinalis. It measures about 3 centimeters in length, about 2 centimeters in breadth at its widest part, and about 1.25 centimeters in thickness. The central canal of the medulla spinalis is prolonged into its lower half and then opens into the cavity of the fourth ventricle. The medulla oblongata may therefore be divided into a lower, closed part containing the central canal and an upper, open part corresponding with the lower portion of the fourth ventricle. The anterior median fissure, fissura mediana anterior, ventral or ventromedian fissure, contains a fold of pia mater and extends along the entire length of the medulla oblongata. It ends at the lower border of the pons in a small triangular expansion termed the foramen cecum. Its lower part is interrupted by bundles of fibers which cross obliquely from one side to the other and constitute the pyramidal decussation. Some fibers, termed the anterior external arcuate fibers, emerge from the fissure above this decussation and curve lateralward and upward over the surface of the medulla oblongata to join the inferior peduncle. The posterior median fissure, fissura mediana posterior, dorsal or dorsal-medial fissure, is a narrow groove and exists only in the closed part of the medulla oblongata. It becomes gradually shallower from below upward and finally ends about the middle of the medulla oblongata, where the central canal expands into the cavity of the fourth ventricle. These two fissures divide the close part of the medulla oblongata into symmetrical halves, each presenting elongated eminences which, on the surface view, are continuous with the funiculi of the medulla spinalis. In the open part, the halves are separated by the anterior median fissure and by a median raphe, which extends from the bottom of the fissure to the floor of the fourth ventricle. Further, certain of the cranial nerves pass through the substance of the medulla oblongata and are attached to its surface in series with the roots of the spinal nerves. Thus, the fibers of the hypoglossal nerve represent the upward continuation of the anterior nerve roots and emerge in linear series from a furrow termed the anterior lateral sulcus. Similarly, the accessory, vagus, and glossopharyngeal nerves correspond with the posterior nerve roots and are attached to the bottom of a sulcus named the posterior lateral sulcus. Advantage is taken of this arrangement to subdivide each half of the medulla oblongata into three districts: anterior, middle, and posterior. Although these three districts appear to be directly continuous with the corresponding funiculi of the medulla spinalis, they do not necessarily contain the same fibers, since some of the fasciculi of the medulla spinalis end in the medulla oblongata, while others alter their course in passing through it. The anterior district is named the pyramid, pyramis medulla oblongata, and lies between the anterior median fissure and the anterior lateral sulcus. Its upper end is slightly constricted, and between it and the pons the fibers of the abducent nerve emerge. A little below the pons it becomes enlarged and prominent, and finally tapers into the anterior funiculus of the medulla spinalis, with which, at first sight, it appears to be directly continuous. The two pyramids contain the motor fibers which pass from the brain to the medulla oblongata and medulla spinalis, corticobulbar and corticospinal fibers. When these pyramidal fibers are traced downward, it is found that some 2/3 or more of them leave the pyramids in successive bundles and decussate in the anterior median fissure, forming what is termed the pyramidal decussation. Having crossed the middle line, they pass down in the posterior part of the lateral funiculus as the lateral cerebrospinal fasciculus. The remaining fibers, that is, those which occupy the lateral part of the pyramid, do not cross the middle line, but are carried downward as the anterior cerebrospinal fasciculus into the anterior funiculus of the same side. The greater part of the anterior proper fasciculus of the medulla spinalis is continued upward through the medulla oblongata under the name of the medial-longitudinal fasciculus. The lateral district is limited in front by the anterior lateral sulcus and the roots of the hypoglossal nerve, and behind by the posterior lateral sulcus and the roots of the accessory, vagus, and glossopharyngeal nerves. Its upper part consists of a prominent oval mass which is named the olive, while its lower part is of the same width as the lateral funiculus of the medulla spinalis and appears on the surface to be a direct continuation of it. As a matter of fact, only a portion of the lateral funiculus has continued upward into this district, for the lateral cerebrospinal fasciculus passes into the pyramid of the opposite side, and the dorsal spinocerebellar fasciculus is carried into the inferior peduncle in the posterior district. The ventral spinocerebellar fasciculus is continued upward on the lateral surface of the medulla oblongata in the same relative position it occupies in the spinal cord, until it passes under cover of the external arcuate fibers. It passes beneath these fibers, just dorsal to the olive and ventral to the roots of the vagus and glossopharyngeal nerves. It continues upward through the pons along the dorsolateral edge of the lateral lemniscus. The remainder of the lateral funiculus consists chiefly of the lateral proper fasciculus. Most of these fibers dip beneath the olive and disappear from the surface, but a small strand remains superficial to the olive. In a depression at the upper end of this strand is the acoustic nerve. The olive, oliva, olivary body, is situated lateral to the pyramid, from which it is separated by the anterior lateral sulcus and the fibers of the hypoglossal nerve. Behind, it is separated from the posterior lateral sulcus by the ventral spinocerebellar fasciculus. In the depression between the upper end of the olive and the pons lies the acoustic nerve. It measures about 1.25 centimeters in length, and between its upper end and the pons there is a slight depression to which the roots of the facial nerve are attached. The external arcuate fibers wind across the lower part of the pyramid and olive and enter the inferior peduncle. The posterior district lies behind the posterior lateral sulcus and the roots of the accessory, vagus, and glossopharyngeal nerves, and, like the lateral district, is divisible into a lower and an upper portion. The lower part is limited behind by the posterior median fissure and consists of the fasciculus gracilis and the fasciculus cuneatus. The fasciculus gracilis is placed parallel to and along the side of the posterior median fissure, and separated from the fasciculus cuneatus by the posterior intermediate sulcus and septum. The gracile and cuneate fasciculi are at first vertical in direction, but at the lower part of the rhomboid fossa they diverge from the middle line in a V-shaped manner, and each presents an elongated swelling. That on the fasciculus gracilis is named the clava, and is produced by its subjacent nucleus of gray matter, the nucleus gracilis. That on the fasciculus cuneatus is termed the cuneate tubercle, and is likewise caused by a gray nucleus named the nucleus cuneatus. The fibers of these fasciculi terminate by arborizing around the cells in their respective nuclei. A third elevation, produced by the substantia gelatinosus of Rolando, is present in the lower part of the posterior district of the medulla oblongata. It lies on the lateral aspect of the fasciculus cuneatus and is separated from the surface of the medulla oblongata by a band of nerve fibers which form the spinal tract, spinal root, of the trigeminal nerve. Narrow below, this elevation gradually expands above and ends about 1.25 centimeters below the pons in a tubercle, the tubercle of Rolando, tuber cinereum. The upper part of the posterior district of the medulla oblongata is occupied by the inferior peduncle, a thick rope-like strand situated between the lower part of the fourth ventricle and the roots of the glossopharyngeal and vagus nerves. The inferior peduncles connect the medulla spinalis and medulla oblongata with the cerebellum and are sometimes named the restiform bodies. As they pass upward, they diverge from each other and assist in forming the lower part of the lateral boundaries of the fourth ventricle. Higher up, they are directed backwards, each passing to the corresponding cerebellar hemisphere. Near their entrance into the cerebellum, they are crossed by several strands of fibers which run to the median sulcus of the rhomboid fossa and are named the stria medullaris. The inferior peduncle appears to be the upward continuation of the fasciculus gracilis and fasciculus cuneatus. This, however, is not so, as the fibers of these fasciculi end in the gracile and cuneate nuclei. The constitution of the inferior peduncle will be subsequently discussed. Caudal to the stria medullaris, the inferior peduncle is partly covered by the corpus pontobulbar, a thin mass of cells and fibers extending from the pons between the origin of the 7th and 8th cranial nerves. Internal structure of the medulla oblongata. Although the external form of the medulla oblongata bears a certain resemblance to that of the upper part of the medulla spinalis, its internal structure differs widely from that of the latter, and this for the following principal reasons. 1. Certain fasciculi, which extend from the medulla spinalis to the brain and vice versa, undergo a rearrangement in their passage through the medulla oblongata. 2. Others, which exist in the medulla spinalis, end in the medulla oblongata. 3. New fasciculi originate in the gray substance of the medulla oblongata and pass to different parts of the brain. 4. The gray substance, which in the medulla spinalis forms a continuous H-shaped column, becomes greatly modified and subdivided in the medulla oblongata, where also new masses of gray substance are added. 5. On account of the opening out of the central canal of the medulla spinalis, certain parts of the gray substance, which in the medulla spinalis were more or less centrally situated, are displaced in the rhomboid fossa. 6. The medulla oblongata is intimately associated with many of the cranial nerves, some arising from and others ending in nuclei within its substance. The cerebrospinal fasciculi. The downward course of these fasciculi from the pyramids of the medulla oblongata and their partial decussation have already been described. In crossing to reach the lateral funiculus of the opposite side, the fibers of the lateral cerebrospinal fasciculi extend backwards through the anterior columns and separate the head of each of these columns from its base. The base retains its position in relation to the ventral aspect of the central canal, and when the latter opens into the fourth ventricle, appears in the rhomboid fossa close to the middle line, where it forms the nuclei of the hypoglossal and abducent nerves, while above the level of the ventricle, it exists as the nuclei of the trochlear and oculomotor nerves in relation to the floor of the cerebral aqueduct. The head of the column is pushed lateralward and forms the nucleus ambiguus, which give origin from below upward to the cranial part of the accessory and the motor fibers of the vagus and glossopharyngeal, and still higher to the motor fibers of the facial and trigeminal nerves. The fasciculus gracilis and fasciculus cuneatus constitute the posterior sensory fasciculi of the medulla spinalis. They are prolonged upward into the lower part of the medulla oblongata, where they end respectively in the nucleus gracilis and nucleus cuneatus. These two nuclei are continuous with the central gray substance of the medulla spinalis and may be regarded as dorsal projections of this, each being covered superficially by the fibers of the corresponding fasciculus. On transverse section, the nucleus gracilis appears as a single, more or less quadrangular mass, while the nucleus cuneatus consists of two parts, a larger, somewhat triangular medial nucleus composed of small or medium-sized cells, and a small lateral nucleus containing large cells. The fibers of the fasciculus gracilis and fasciculus cuneatus end by arborizing around the cells of these nuclei. From the cells of the nuclei, new fibers arise. Some of these are continued as the posterior external arcuate fibers into the inferior peduncle and through it to the cerebellum, but most of them pass forward through the neck of the posterior column, thus cutting off its head from its base. Curving forward, they decussate in the middle line with the corresponding fibers of the opposite side and run upward immediately behind the cerebrospinal fibers as a flattened band named the lemniscus or fillet. The decussation of these sensory fibers is situated above that of the motor fibers and is named the decussation of the lemniscus or sensory decussation. The lemniscus is joined by the spinothalamic fasciculus, the fibers of which are derived from the cells of the gray substance of the opposite side of the medulla spinalis. The base of the posterior column at first lies on the dorsal aspect of the central canal, but when the latter opens into the fourth ventricle, it appears in the lateral part of the rhomboid fossa. It forms the terminal nuclei of the sensory fibers of the vagus and glossopharyngeal nerves and is associated with the vestibular part of the acoustic nerve and the sensory root of the facial nerve. Still higher, it forms a mass of pigmented cells, the locus ceruleus, in which some of the sensory fibers of the trigeminal nerve appear to end. The head of the posterior column forms a long nucleus, in which the fibers of the spinal tract of the trigeminal nerve largely end. The dorso-spinocerebellar fasciculus, fasciculus cerebellospinalis, direct cerebellar tract, leaves the lateral district of the medulla oblongata. Most of its fibers are carried backward into the inferior peduncle of the same side, and through it are conveyed to the cerebellum. But some run upward with the fibers of the lemniscus and, reaching the inferior colliculus, undergo decussation and are carried to the cerebellum through the superior peduncle. The proper fasciculi, basis bundles, of the anterior and lateral funiculi largely consist of intersegmental fibers, which link together the different segments of the medulla spinalis. They assist in the production of the famicula reticularis in the medulla oblongata, and many of them are accumulated into a fasciculus which runs up close to the median raphe between the lemniscus and the rhomboid fossa. This strand is named the medial longitudinal fasciculus and will be again referred to. End of section 8. Section 9 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Hindbrain Part 2. Gray substance of the medulla oblongata. In addition to the gracile and cuneate nuclei, there are several other nuclei to be considered. Some of these are traceable from the gray substance of a medulla spinalis, while others are unrepresented in it. 1. The hypoglossal nucleus is derived from the base of the anterior column. In the lower closed part of the medulla oblongata, it is situated on the ventrolateral aspect of the central canal, but in the upper part, it approaches the rhomboid fossa, where it lies close to the middle line under an eminence named the trigonum hypoglossae. Numerous fibers connect the two nuclei. Both nuclei send long dendrons across the midline to the opposite nucleus. Commissure fibers also connect them. The nucleus measures about 2 centimeters in length and consists of large multipolar nerve cells similar to those in the anterior column of the spinal cord, whose axons constitute the roots of the hypoglossal nerve. These nerve roots leave the ventral side of the nucleus, pass forward between the white reticular formation and the gray reticular formation, some between the inferior olivary nucleus and the medial accessory olivary nucleus, and emerge from the anterolateral sulcus. 2. The nucleus ambiguus, the somatic motor nucleus of the glossopharyngeal, vagus, and cranial portion of the accessory nerves, is the continuation into the medulla oblongata of the dorsolateral cell group of the anterior column of the spinal cord. Its large, multipolar cells are like those in the anterior column of the cord. They form a slender column in the deep part of the formatio reticularis grisae, about midway between the dorsal accessory olive and the nucleus of the spinal tract of the trigeminal. It extends from the level of the decussation of the median fillet to the upper end of the medulla oblongata. Its fibers first pass backward towards the floor of the fourth ventricle, and then curve rather abruptly lateralward and ventrally to join the fibers from the dorsal nucleus. The dorsal nucleus, nucleus allocenaria, often called the sensory nucleus or the trigeminal nucleus of the sensory fibers of the glossopharyngeal and vagus nerves, is probably a mixed nucleus and contains not only the terminations of the sympathetic afferent or sensory fibers and the cells connected with them, but contains also cells which give rise to the sympathetic efferent or preganglionic fibers. These preganglionic fibers terminate in sympathetic ganglia, from which the impulses are carried by other neurons. The cells of the dorsal nucleus are spindle-shaped like those of the posterior column of the spinal cord, and the nucleus is usually considered as representing the base of the posterior column. It measures about 2 centimeters in length, and in the lower, closed part of the medulla oblongata, is situated behind the hypoglossal nucleus, whereas in the upper open part, it lies lateral to that nucleus and corresponds to an eminence named the allocenaria, trigonum vagi, in the rhomboid fossa. 4. The nuclei of the cochlear and vestibular nerves are described later. 5. The olivary nuclei are 3 in number on either side of the midline, that is, the inferior olivary nucleus, and the medial and dorsal accessory olivary nuclei. They consist of small round yellowish cells and numerous fine nerve fibers. A. The inferior olivary nucleus is the largest and is situated within the olive. It consists of a gray folded lamina arranged in the form of an incomplete capsule, open medially by an aperture called the hilum. Emerging from the hilum are numerous fibers which collectively constitute the peduncle of the olive. The axons, olivocerebellar fibers, which leave the olivary nucleus, pass out through the hilum and decussate with those from the opposite olive in the raphe. Then, as internal arcuate fibers, they pass partly through and partly around the opposite olive and enter the inferior peduncle to be distributed to the cerebellar hemisphere of the opposite side from which they arise. The fibers are smaller than the internal arcuate fibers connected with the medial lemniscus. Fibers passing in the opposite direction from the cerebellum to the olivary nucleus are often described, but their existence is doubtful. Much uncertainty also exists in regard to the connections of the olive and the spinal cord. Important connections between the cerebellum and the olive of the same side exist, but the exact pathway is unknown. Many collaterals from the reticular formation and from the pyramids enter the inferior olivary nucleus. Removal of one cerebellar hemisphere is followed by atrophy of the opposite olivary nucleus. B. The medial accessory olivary nucleus lies between the inferior olivary nucleus and the pyramid and forms a curved lamina, the concavity of which is directed laterally. The fibers of the hypoglossal nerve, as they traverse the medulla, pass between the medial accessory and the inferior olivary nuclei. C. The dorsal accessory olivary nucleus is the smallest and appears on transverse section as the curved lamina behind the inferior olivary nucleus. 6. The nucleus arcuatus is described below with the anterior external arcuate fibers. Inferior peduncle, restiform body. The position of the inferior peduncles has already been described. Each comprises 1. Fibers from the dorsal spinocerebellar fasciculus, which ascends from the lateral funiculus of the medulla spinalis. 2. The olivocerebellar fibers from the opposite olivary nucleus. 3. Internal arcuate fibers from the gracillin cuneate nuclei of the opposite side. These fibers form the deeper and larger part of the inferior peduncle. 4. The anterior external arcuate fibers vary as to the prominence in different cases. In some, they form an almost continuous layer covering the pyramid and olive, while in others they are barely visible on the surface. They arise from the cells of the gracillin cuneate nuclei and, passing forward through the formatio reticularis, decussate in the middle line. Most of them reach the surface by way of the anterior median fissure and arch backward over the pyramid. Reinforced by others which emerge between the pyramid and olive, they pass backwards over the olive and lateral district of the medulla oblongata and enter the inferior peduncle. Thus, they connect the cerebellum with the gracillin cuneate nuclei of the opposite side. As the fibers arch across the pyramid, they enclose a small nucleus which lies in front of and medial to the pyramid. This is named the nucleus arcuatus, and is serially continuous above with the nucleus pontus and the pons. It contains small fusiform cells around which some of the arcuate fibers end and from which others arise. 5. The posterior external arcuate fibers also take origin in the gracillin cuneate nuclei. They pass to the inferior peduncle of the same side. It is uncertain whether fibers are continued directly from the gracillin cuneate fasciculi into the inferior peduncle. 6. Fibers from the terminal sensory nuclei of the cranial nerves, especially the vestibular. Some of the fibers of the vestibular nerve are thought to continue directly into the cerebellum. 7. Fibers from the ventral spinocerebellar fasciculus. 8. The existence of fibers from the cerebellum, cerebellobulbar, cerebellar olivary, and cerebellar spinal, to the medulla and spinal cord is very uncertain. Formatio reticularis. This term is applied to the coarse reticulum which occupies the anterior and lateral districts of the medulla oblongata. It is situated behind the pyramid and olive, extending laterally as far as the inferior peduncles, and dorsally to within a short distance of the rhomboid fossa. The reticulum is caused by the intersection of bundles of fibers running at right angles to each other, some being longitudinal, others more or less transverse in direction. The formatio reticularis presents a different appearance in the anterior district from what it does in the lateral. In the former, there is an almost entire absence of nerve cells, and hence this part is known as the reticularis alba, whereas in the lateral district nerve cells are numerous, and as a consequence it presents a gray appearance and is termed the reticularis grisea. In the substance of the formatio reticularis are two small nuclei of gray matter. One, the inferior central nucleus, nucleus of Roller, near the dorsal aspect of the hilus of the inferior olivary nucleus. The other, the nucleus lateralis, between the olive and the spinal tract of the trigeminal nerve. In the reticularis alba, the longitudinal fibers form two well-defined fasciculi. That is, 1, the lemniscus, which lies close to the raphe immediately behind the fibers of the pyramid, and 2, the medial longitudinal fasciculus, which is continued upward from the anterior and lateral proper fasciculi of the medulla spinalis, and, in the upper part of the medulla oblongata, lies between the lemniscus and the gray substance of the rhomboid fossa. The longitudinal fibers in the reticularis grisea are derived from the lateral funiculus of the medulla spinalis after the lateral cerebrospinal fasciculus has passed over to the opposite side, and the dorsal spinocerebellar fasciculus has entered the inferior peduncle. They form indeterminate fibers, with the exception of a bundle named the fasciculus solitarius, which is made up of descending fibers of the vagus and glossopharyngeal nerves. The transverse fibers of the formatio reticularis are the arcuate fibers already described. The pons. Pons varioli. The pons, or forepart of the hindbrain, is situated in front of the cerebellum. From its superior surface, the cerebral peduncles emerge, one on either side of the middle line. Curving around each peduncle, close to the upper surface of the pons, a thin white band, the tinea pontis, is frequently seen. It enters the cerebellum between the middle and superior peduncles. Behind and below, the pons is continuous with the medulla oblongata, but is separated from it in front by a furrow in which the abducent, facial, and acoustic nerves appear. Its ventral or anterior surface, pars basilaris pontis, is very prominent, markedly convex from side to side, less so from above downward. It consists of transverse fibers arched like a bridge across the middle line and gathered on either side into a compact mass which forms the middle peduncle. It rests upon the clivus of the sphenoidal bone and is limited above and below by well-defined borders. In the middle line is the sulcus basilaris for the lodgment of the basilar artery. This sulcus is bounded on either side by an eminence caused by the descent of the cerebrospinal fibers through the substance of the pons. Outside these eminences, near the upper border of the pons, the trigeminal nerves make their exit, each consisting of a smaller mediomotor root and a larger lateral sensory root. Vertical lines drawn immediately beyond the trigeminal nerves may be taken as the boundaries between the ventral surface of the pons and the middle cerebellar peduncle. Its dorsal or posterior surface, pars dorsalis pontis, triangular in shape, is hidden by the cerebellum and is bounded laterally by the superior peduncle. It forms the upper part of the rhomboid fossa, with which it will be described. Structure. Transverse sections of the pons show it to be composed of two parts, which differ in appearance and structure. Thus, the basilar or ventral portion consists for the most part of fibers arranged in transverse and longitudinal bundles, together with a small amount of gray substance, while the dorsal tegmental portion is a continuation of the reticular formation of the medulla oblongata, and most of its constituents are continued into the tegmenta of the cerebral peduncles. The basilar part of the pons consists of A. Superficial and deep transverse fibers, B. Longitudinal fasciculi, and C. Some small nuclei of gray substance, termed the nuclei pontis, which give rise to the transverse fibers. The superficial transverse fibers, fibrae pontis superficialis, constitute a rather thick layer on the ventral surface of the pons and are collected into a large rounded bundle on either side of the middle line. This bundle, with the addition of some transverse fibers from the deeper part of the pons, forms the greater part of the brachium pontis. The deep transverse fibers, fibrae pontis profundae, partly intersect and partly lie in the dorsal aspect of the cerebrospinal fibers. They course to the lateral border of the pons and form part of the middle peduncle. A further connections of this brachium will be discussed with the anatomy of the cerebellum. The longitudinal fasciculi longitudinalis, are derived from the cerebral peduncles and enter the upper surface of the pons. They stream downward on either side of the middle line in larger or smaller bundles separated from each other by the deep transverse fibers. These longitudinal bundles cause a forward projection of the superficial transverse fibers and thus give rise to the eminences on the anterior surface. Some of these fibers end in or give off collateral to the nuclei pontis. An important pathway is thus formed between the cerebral cortex and the cerebellum, the first neuron having its cell body in the cortex and sending its axon through the internal capsule and cerebral peduncle to form synapses either by terminals or collaterals with cell bodies situated in the nuclei pontis. Axons from these cells form the transverse fiber which pass through the middle peduncle into the cerebellum. Others, after decussating, terminate either directly or indirectly in the motor nuclei of the trigeminal, abducent, facial, and hypoglossal nerves, but most of them are carried through the pons and at the lower surface are collected into the pyramids of the medulla. The fibers which end in the motor nuclei of the cranial nerves are derived from the cells of the cerebral cortex and bear the same relation to the motor cells of the cranial nerves that the cerebrospinal fibers bear to the motor cells in the anterior column of the medulla spinalis. Probably none of the collaterals or terminals of the cerebrospinal and cerebrobulbar fibers end directly in the motor nuclei of the spinal and cranial nerves. One or more association neurons are probably interpolated in the pathway. The nuclei pontis are serially continuous with the arcuate nuclei in the medulla and consist of small groups of multipolar nerve cells which are scattered between the bundles of the transverse fibers. The dorsal or tegmental part of the pons is chiefly composed of an upward continuation of the reticular formation and gray substance of the medulla oblongata. It consists of transverse and longitudinal fibers and also contains important gray nuclei and is subdivided by a median raphe which, however, does not extend into the basilar part, being obliterated by the transverse fibers. The transverse fibers in the lower part of the pons are collected into a distinct strand named the trapezoid body. This consists of fibers which arise from cells of the cochlear nucleus and will be referred to in connection with the cochlear division of the acoustic nerve. In the substance of the trapezoid body is a collection of nerve cells which constitutes the trapezoid nucleus. The longitudinal fibers, which are continuous with those of the medulla oblongata, are mostly collected into two fasciculae on each side. One of these lies between the trapezoid body and the reticular formation and forms the upward prolongation of the lemniscus. The second is situated near the floor of the fourth ventricle and is the medial longitudinal fasciculus. Other longitudinal fibers, more diffusely distributed, arise from the cells of the gray substance of the pons. The rest of the dorsal part of the pons is a continuation upward of the formatio reticularis of the medulla oblongata, and like it, presents the appearance of a network, in the meshes of which are numerous nerve cells. Besides these scattered nerve cells, there are some larger masses of gray substance, that is, the superior olivary nucleus and the nuclei of the trigeminal, abducent, facial, and acoustic nerves. 1. The superior olivary nucleus, nucleus olivaris superior, is a small mass of gray substance situated on the dorsal surface of the lateral part of the trapezoid body. Rudimentary in man, but well developed in certain animals, it exhibits the same structure as the inferior olivary nucleus and is situated immediately above it. Some of the fibers of the trapezoid body end by arborizing around the cells of this nucleus, while others arise from these cells. 2. The nuclei of the trigeminal nerve. Nuclei and trigemini. In the pons are two in number, a motor and a sensory. The motor nucleus is situated in the upper part of the pons, close to its posterior surface and along the line of the lateral margin of the fourth ventricle. It is serially homologous with the nucleus ambiguus and the dorsolateral cell group of the anterior column of the spinal cord. The axis cylinder processes of its cells form a portion of the motor root of the trigeminal nerve. The remaining fibers of the motor root of this nerve consist of a fasciculus, which arises from the gray substance of the floor of the cerebral aqueduct, and hence is named the mesencephalic root. It is not altogether clear whether the mesencephalic root is motor or sensory. The sensory nucleus is lateral to the motor one and beneath the superior peduncle. Some of the sensory fibers of the trigeminal nerve end in this nucleus, but the greater number descend under the name of the spinal tract of the trigeminal nerve to end in the substantia gelatinosa of Rolando. The roots, motor and sensory, of the trigeminal nerve pass through the substance of the pons and emerge near the upper margin of its anterior surface. 3. The nucleus of the abducent nerve, nucleus n abducentus, is a circular mass of gray substance situated close to the floor of the fourth ventricle, above the striae medullaris and subjacent to the medial eminence. It lies a little lateral to the ascending part of the facial nerve. The fibers of the abducent nerve pass forward through the entire thickness of the pons on the medial side of the superior olivary nucleus and between the lateral fasciculi of the cerebrospinal fibers, and emerge in the furrow between the lower border of the pons and the pyramid of the medulla oblongata. The nucleus of the facial nerve, nucleus n facialis, is situated deeply in the reticular formation of the pons, on the dorsal aspect of the superior olivary nucleus, and the roots of the nerve derived from it pursue a remarkably tortuous course in the substance of the pons. At first, they pass backward and medialward until they reach the rhomboid fossa, close to the median sulcus, where they are collected into a round bundle. This passes upward and forward, producing an elevation, the colliculus facialis, in the rhomboid fossa, and then takes a sharp bend and arches lateralward through the substance of the pons to emerge at its lower border in the interval between the olive and the inferior peduncle of the medulla oblongata. 5. The nucleus of the cochlear nerve consists of a. The laterocochlear nucleus, corresponding to the tuberculum acousticum on the dorsolateral surface of the inferior peduncle, and b. The ventral or exsensory cochlear nucleus, placed between the two divisions of the nerve on the ventral aspect of the inferior peduncle. The nuclei of the vestibular nerve. A. The medial, dorsal or chief vestibular nucleus, corresponding to the lower part of the area acoustica in the rhomboid fossa. The caudal end of this nucleus is sometimes termed the descending or spinal vestibular nucleus. B. The lateral, or nucleus of Dieters, consisting of large cells and situated in the lateral angle of the rhomboid fossa. The dorsolateral part of this nucleus is sometimes termed the nucleus of Bechtereau. The fibers of the vestibular nerve enter the medulla oblongata on the medial side of those of the cochlear and pass between the inferior peduncle and the spinal tract of the trigeminal. They then divide into ascending and descending fibers. The latter end by arborizing around the cells of the medial nucleus, which is situated in the area acoustica of the rhomboid fossa. The ascending fibers either end in the same manner or in the lateral nucleus, which is situated lateral to the area acoustica and farther from the ventricular floor. Some of the axons of the cells of the lateral nucleus, and possibly also the medial nucleus, are continued upward through the inferior peduncle to the roof nuclei of the opposite side of the cerebellum, to which also other fibers of the vestibular root are prolonged without interruption in the nuclei of the medulla oblongata. A second set of fibers from the medial and lateral nuclei and partly in the tegmentum, while the remaining ascend in the medial longitudinal fasciculus to arborize around the cells of the nuclei of the ocular motor nerve. End of section 9. Section 10 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Hindbrain Part 3. The cerebellum constitutes the largest part of the hindbrain. It lies behind the pons and medulla oblongata. Between its central portion and these structures is the cavity of the fourth ventricle. It rests on the inferior occipital fossae, while above it is the tentorium cerebellae, a fold of dura mater which separates it from the tentorial surface of the cerebrum. It is somewhat oval in form, but constricted medially and flattened from above downward, its greatest diameter being from side to side. Its surface is not convoluted like that of the cerebrum, but is traversed by numerous curved furrows or sulci, which vary in depth at different parts, and separate the laminae of which it is composed. Its average weight in the male is about 150 grams. In the adult, the proportion between the cerebellum and cerebrum is about 1 to 8. In the infant, about 1 to 20. Lobes of the cerebellum. The cerebellum consists of 3 parts, a median and 2 lateral, which are continuous with each other and are substantially the same in structure. The median portion is constricted and is called the vermis, from its annulated appearance which it owes to the transverse ridges and furrows upon it. The lateral expanded portions are named the hemispheres. On the upper surface of the cerebellum, the vermis is elevated above the level of the hemisphere, but on the undersurface, it is sunk almost out of sight in the bottom of a deep depression between them. This depression is called the vallecula cerebellae and lodges the posterior part of the medulla oblongata. The part of the vermis on the upper surface of the cerebellum is named the superior vermis, that on the lower surface the inferior vermis. The hemispheres are separated below and behind by a deep notch, the posterior cerebellar notch, and in front by a broader, shallower notch, the anterior cerebellar notch. The anterior notch lies close to the pons and upper part of the medulla, and its superior edge encircles the inferior colliculi and the superior cerebellar peduncle. The posterior notch contains the upper part of the falx cerebellae, a fold of dura mater. The cerebellum is characterized by a laminated or foliated appearance. It is marked by deep, somewhat curved fissures, which extend for a considerable distance into its substance and divide it into a series of layers or leaves. The largest and deepest fissure is named the horizontal sulcus. It commences in front of the pons and passes horizontally around the free margin of the hemisphere to the middle line behind and divides the cerebellum into an upper and a lower portion. Several secondary but deep fissures separate the cerebellum into lobes, and these are further subdivided by shallower sulci, which separate the individual folia or laminae from each other. Sections across the laminae show that the folia, though differing in appearance from the convolutions of the cerebrum, are analogous to them inasmuch as they consist of central white substance covered by gray substance. The cerebellum is connected to the cerebrum, pons, and medulla oblongata, to the cerebrum by the superior peduncle, to the pons by the middle peduncle, and to the medulla oblongata by the inferior peduncles. The upper surface of the cerebellum is elevated in the middle and sloped towards the circumference, the hemispheres being connected together by a superior vermis, which assumes the form of a raised median ridge, most prominent in front, but not sharply defined from the hemispheres. The superior vermis is subdivided from before backwards into the lingula, the lobus centralis, the monticulus, and the folium vermis, and each of these, with the exception of the lingula, is continuous with the corresponding parts of the hemispheres, the lobulus centralis with the ali, the monticulus with the quadrigeminal lobules, and the folium vermis with the superior semilunar lobules. The lingula, lingula cerebelli, is a small tongue-shaped process consisting of 4 or 5 folia. It lies in front of the lobulus centralis and is concealed by it. Anteriorly, it rests on the dorsal surface of the anterior medullary velum, and its white substance is continuous with that of the velum. The lobulus centralis and ala. The lobulus centralis is a small square lobule situated in the anterior cerebellar notch. It overlaps the lingula, from which it is separated by the precentral fissure. Laterally, it extends along the upper and anterior part of each hemisphere, where it forms a wing-like prolongation, the ala lobula centralis. The monticulus and quadrangular lobules. The monticulus is the largest part of the superior vermis. Anteriorly, it overlaps the lobulus centralis, from which it is separated by the postcentral fissure. Laterally, it is continuous with the quadrangular lobule in the hemispheres. It is divided by the preclival fissure into an anterior raised part, the culmen or summit, and a posterior slope part, the clivus. The quadrangular lobule is similarly divided. The culmen and the anterior parts of the quadrangular lobules form the lobus culminus, the clivus and the posterior parts the lobus clevi. The folium vermis and superior semilunar lobule. The folium vermis, folium cacuminus, cacuminal lobe, is a short, narrow, concealed band at the posterior extremity of the vermis, consisting apparently of a single folium, but in reality marked on its upper and under surfaces by secondary fissures. Laterally, it expands in either hemisphere into a considerable lobule, the superior semilunar lobule, lobulus semilunaris superior, posterior superior lobules, which occupy the posterior third of the upper surface of the hemispheres, and is bounded below by the horizontal sulcus. The superior semilunar lobules and the folium vermis form the lobus semilunaris. The undersurface of the cerebellum presents in the middle line the inferior vermis, buried in the vallecula and separated from the hemisphere on either side by a deep groove, the sulcus valleculae. Here, as on the upper surface, there are deep fissures dividing it into separate segments or lobules, but the arrangement is more complicated, and the relation of the segments to the vermis to those of the hemispheres is less clearly marked. The inferior vermis is subdivided from below backwards into 1. The nodule, 2. The uvula, 3. The pyramid, and 4. The tumor vermis. The corresponding parts on the hemisphere are 1. The flocculus, 2. The tonsillus cerebelli, 3. The biventral lobule, and 4. The inferior semilunar lobule. The 3 main fissures are 1. The postnodular fissure, which runs transversely across the vermis between the nodule and the uvula. In the hemisphere, this fissure passes in front of the tonsil, crosses between the flocculus in front and the biventral lobule behind, and joins the anterior end of the horizontal sulcus. 2. The prepyramidal fissure crosses the vermis between the uvula in front and the pyramid behind, then curves forward between the tonsil and the biventral lobe to join the postnodular fissure. 3. The postpyramidal fissure passes across the vermis between the pyramid and the tuber vermis and, in the hemispheres, courses behind the tonsil and biventral lobules, and then along the lateral border of the biventral lobule to the postnodular sulcus. In the hemisphere, it forms the anterior boundary of the inferior semilunar lobule. The nodule and flocculus. The nodule, nodulus vermis, nodular lobe, or anterior end of the inferior vermis, abuts against the roof of the fourth ventricle, and can only be distinctly seen after the cerebellum has been separated from the medulla oblongata and pons. On either side of the nodule is a thin layer of white substance named the posterior medullary velum. It is semilunar in form, its convex border being continuous with the white substance of the cerebellum. It extends on either side as far as the flocculus. The flocculus is a prominent irregular lobule situated in front of the biventral lobule, between it and the midcerebellar peduncle. It is subdivided into a few small laminae and is connected to the inferior medullary velum by its central white core. The flocculi, together with the posterior medullary velum and nodule, constitute the lobus noduli. The uvula and tonsilla. The uvula, uvula vermis, uvular lobe, forms a considerable portion of the inferior vermis. It is separated on either side from the tonsil by the sulculus folliculae, at the bottom of which it is connected to the tonsil by a ridge of gray matter, indented on its surface by shallow furrows, and hence called the furrowed band. The tonsilla, tonsilla cerebelli, amygdaline nucleus, is a rounded mass situated in the hemispheres. Each lies in a deep fossa termed the bird's nest, nidus avis, between the uvula and the biventral lobule. The uvula and tonsillae form the lobus uvulae. The pyramid and biventral lobules constitute the lobus pyramidus. The pyramid is a conical portion forming the largest prominence of the inferior vermis. It is separated from the hemispheres by the sulcus folliculae, across which it is connected to the biventral lobule by an indistinct gray band, analogous to the furrowed band already described. The biventral lobule is triangular in shape, its apex points backwards and is joined by the gray band to the pyramid. The lateral border is separated from the inferior semilunar lobule by the postpyramidal fissure. The base is directed forward and is on a line with the anterior border of the tonsil and is separated from the flocculus by the postnodular fissure. The tuber vermis, tuber valvulae, and the inferior semilunar lobule, lobulus semilunaris inferior, posterior superior lobule, collectively form the lobus tuberus, tuberae lobe. The tuber vermis, the most posterior division of the interior vermis, is of small size and laterally spreads out into the large inferior semilunar lobules, which comprise at least 2/3 of the inferior surface of the hemisphere. Internal structure of the cerebellum. The cerebellum consists of white and gray substance. White substance. If a sagittal section be made through either hemisphere, the interior will be found to consist of a central stem of white substance, in the middle of which is a gray mass, the dentate nucleus. From the surface of the central white stem, a series of plates is prolonged. These are covered with gray substance and form the laminae. In consequence of the main branches from the central stem dividing and subdividing, a characteristic appearance named the arbor vitae is presented. If the sagittal section be made through the middle of the vermis, it will be found that the central stem divides into a vertical and horizontal branch. The vertical branch passes upwards to the culmen monticuli, where it subdivides freely, one of its ramifications passing forward and upward to the central lobule. The horizontal branch passes backwards to the folium vermis, greatly diminished in size in consequence of having given off large secondary branches. One, from its upper surface, ascends to the clivus monticuli, the others descend and enter the lobes of the inferior vermis, that is, the tuber vermis the pyramid, the uvula, and the nodule. The white substance of the cerebellum include two sets of nerve fibers. 1. Projection fibers. 2. Fibrae propriae. Projection fibers. The cerebellum is connected to other parts of the brain by 3 large bundles of projection fibers. That is, to the cerebrum by the superior peduncle, to the pons by the middle peduncle, and to the medulla oblongata by the inferior peduncles. The superior cerebellar peduncles, brachia conjunctiva, 2 in number, emerge from the upper and medial portions of the white substance of the hemispheres and are placed under cover of the upper part of the cerebellum. They are joined to each other across the middle line by the anterior medullary velum and can be followed upward as far as the inferior colliculi, under which they disappear. Below, they form the upper lateral boundaries of the fourth ventricle, but as they ascend, they converge on the dorsal aspect of the ventricle and thus assist in roofing it in. The fibers of the superior peduncle are mainly derived from the cells of the dentate nucleus of the cerebellum and emerge from the hilus of this nucleus. A few arise from the cells of the smaller gray nuclei in the cerebellar white substance, and others from the cells of the cerebellar cortex. They are continued upward beneath the corpora quadrigemina, and the fibers of the 2 peduncles undergo a complete decussation ventral to the sylvian aqueduct. Having crossed the middle line, they divide into ascending and descending groups of fibers, the former ending in the red nucleus, the thalamus, and the nucleus of the ocular motor nerve, while the descending fibers can be traced as far as the dorsal part of the pons. Cajal believes them to be continued into the anterior funiculus of the medulla spinalis. As already stated, the majority of the fibers of the ventral spinocerebellar fasciculus of the medulla spinalis pass to the cerebellum, which they reach by way of the superior peduncle. The middle cerebellar peduncles, brachia pontis, are composed entirely of centripetal fibers which arise from the cells of the nuclei pontis of the opposite side and end in the cerebellar cortex. The fibers are arranged in 3 fasciculi, superior, inferior, and deep. The superior fasciculus, the most superficial, is derived from the upper transverse fibers of the pons. It is directed backward and lateralward superficial to the other 2 fasciculi and is distributed mainly to the lobules on the inferior surface of the cerebellar hemisphere and to the parts of the superior surface adjoining the posterior and lateral margins. The inferior fasciculus is formed by the lowest transverse fibers of the pons. It passes under cover of the superior fasciculus and continued downward and backward more or less parallel with it to be distributed to the folia on the undersurface close to the vermis. The deep fasciculus comprises most of the deep transverse fibers of the pons. It is at first covered by the superior and inferior fasciculi, but crosses obliquely and appears on the medial side of the superior, from which it receives a bundle. Its fibers spread out and pass to the upper anterior cerebellar folia. The fibers of this fasciculus cover those of the restiform body. The inferior cerebellar peduncles, restiform bodies, pass at first upward and lateralward, forming part of the lateral walls of the fourth ventricle, and then bend abruptly backwards to enter the cerebellum between the superior and middle peduncles. Each contains the following fasciculi. 1. The dorsal spinocerebellar fasciculus of the medulla spinalis, which ends mainly in the superior vermis. 2. Fibers from the gracillin cuneate nuclei of the same side and of the opposite sides. 3. Fibers from the opposite olivary nuclei. 4. Crossed and uncrossed fibers from the reticular formation of the medulla oblongata. 5. Vestibular fibers, derived partly from the vestibular division of the acoustic nerve and partly from the nuclei in which this division ends. These fibers occupy the medial segment of the inferior peduncle and divide into ascending and descending groups of fibers. The ascending fibers partly end in the roof nucleus on the opposite side of the cerebellum. 6. Cerebellobulbar fibers, which come from the opposite roof nucleus, and probably from the dentate nucleus, and are said to end in the nucleus of Dieters and in the formatio reticularis of the medulla oblongata. 7. Some fibers from the ventral spinocerebellar fasciculus are said to join the dorsal spinocerebellar fasciculus. The anterior medullary velum. Velum medullare anterius, valve of Vincennes, superior medullary velum, is a thin, transparent lamina of white substance which stretches between the superior peduncle on the dorsal surface of its lower half. The folia and lingula are prolonged. It forms, together with the superior peduncle, the roof of the upper part of the fourth ventricle. It is narrow above, where it passes beneath the inferior colliculi, and broader below, where it is continuous with the white substance of the superior vermis. A slightly elevated ridge, the frenulum veli, descends upon its upper part from between the inferior colliculi, and on either side of this the trochlear nerve emerges. The posterior medullary velum, velum medullare posterius, inferior medullary velum, is a thin layer of white substance prolonged from the white center of the cerebellum above and on either side of the nodule. It forms a part of the roof of the fourth ventricle. Somewhat semilunar in shape, its convex edge is continuous with the white substance of the cerebellum, while its thin concave margin is apparently free. In reality, however, it is continuous with the epithelium of the ventricle, which is prolonged downward from the posterior medullary velum to the ligulae. The two medullary vela are in contact with each other along their line of emergence from the white substance of the cerebellum, and this line of contact forms the summit of the roof of the fourth ventricle, which, in a vertical section through the cavity, appears as a pointed angle. The fibrae propriae of the cerebellum are of two kinds. One, commissural fibers, which cross the middle lines at the anterior and posterior parts of the vermis and connect the opposite halves of the cerebellum. Two, arcuate or association fibers, which connect adjacent laminae with each other. Gray substance. The gray substance of the cerebellum is found in two situations. 1. On the surface forming the cortex. 2. As independent masses in the anterior. 1. The gray substance of the cortex presents a characteristic foliated appearance due to the series of laminae which are given off from the central white substance. These, in turn, give off secondary laminae which are covered by gray substance. Externally, the cortex is covered by pia mater. Internally is the medullary center, consisting mainly of nerve fibers. End of section 10. Section 11 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. The Hindbrain, Part 4. Microscopic appearance of the cortex. The cortex consists of two layers, that is, an external gray molecular layer and an internal rust-colored nuclear layer. Between these is an incomplete stratum of cells which are characteristic of the cerebellum, that is, the cells of Purkinje. The external gray or molecular layer consists of fibers and cells. The nerve fibers are delicate fibrillae and are derived from the following sources. A. The dendrites and axon collaterals of Purkinje cells. B. Fibers from the cells of the nuclear layer. C. Fibers from the central white substance of the cerebellum. D. Fibers derived from cells in the molecular layer itself. In addition to these are other fibers, which have a vertical direction and are the processes of the large neuroglial cells situated in the nuclear layer. They pass outward to the periphery of the gray matter, where they expand into little conical enlargements, which form a sort of limiting membrane beneath the pia mater, analogous to the membrana limitans interna in the retina, formed by the sustentacular fibers of Mueller. The cells of the molecular layer are small and are arranged in two striata, an outer and an inner. They all possess branched axons, Those of the inner layer are termed basket cells. They run for some distance parallel with the surface of the folium, giving off collaterals which pass in a vertical direction towards the bodies of the Purkinje cells, around which they become enlarged and form basket-like networks. The cells of Purkinje form a single stratum of large flask-like shells at the junction of the molecular and nuclear layers, their bases resting against the latter. In fishes and reptiles, they are arranged in several layers. The cells are flattened in a direction transverse to the long axis of the folium, and thus appear broad in sections carried across the folium, and fusiform in sections parallel to the long axis of the folium. From the neck of the flask, one or more dendrites arise and pass into the molecular layer, where they subdivide and form an extremely rich arborescence, the various subdivisions of the dendrites being covered by lateral spine-like processes. The arborescence is not circular, but, like the cell, is flattened at right angles to the long axis of the folium. In other words, it does not resemble a round bush, but has been aptly compared by Obersteiner to the branches of a fruit tree trained against a trellis or a wall. Hence, in sections carried across the folium, the arborescence is broad and expanded, where in those which are parallel to the long axis of the folium, the arborescence, like the cell itself, is seen in profile and is limited to a narrow area. From the bottom of the flask-shaped cell, the axon arises. This passes through the nuclear layer and, becoming medullated, is continued as a nerve fiber in the subjacent white substance. As this axon traverses the granular layer, it gives off fine collaterals, some of which run back into the molecular layer. The internal, rust-colored or nuclear layer is characterized by containing numerous small nerve cells of a reddish-brown color, together with many nerve fibrils. Most of the cells are nearly spherical and provided with short dendrites which spread out in a spider-like manner in the nuclear layer. Their axons pass outward into the molecular layer and, bifurcating at right angles, run for some distance parallel with the surface. In the outer part of the nuclear layer are some larger cells of the type 2 of Golgi. Their axons undergo frequent division as soon as they leave the nerve cells and pass into the nuclear layer, while their dendrites ramify chiefly in the molecular layer. Finally, in the gray substance of the cerebellar cortex, there are fibers which come from the white center and penetrate the cortex. The cell origin of these fibers is unknown, though it is believed that it is probably in the gray substance of the medulla spinalis. Some of these fibers end in the nuclear layer by dividing into numerous branches, on which are to be seen peculiar moss-like appendages. Hence, they have been termed by Raymond y Cajal the moss fibers. They form an arborescence around the cells of the nuclear layer and are said to come from fibers in the inferior peduncle. Other fibers, the clinging or tendril fibers, derived from the medullary center, can be traced into the molecular layer, where their branches cling around the dendrites of Purkinje cells. They are said to come from fibers of the middle peduncle. 2. The independent centers of gray substance in the cerebellum are 4 in number on either side. One is of large size and is known as the nucleus dentatus. The other 3, much smaller, are situated near the middle of the cerebellum and are known as the nucleus embryoloformis, nucleus globosus, and nucleus fastigii. The nucleus dentatus is situated a little to the medial side of the center of the stem of the white substance of the hemisphere. It consists of an irregularly folded lamina of grayish-yellow color, containing white fibers, and presenting on its anterior medial aspect an opening, the hilus, from which most of the fibers of the superior peduncle emerge. The nucleus embryoloformis lies immediately to the medial side of the nucleus dentatus and partly covering its hilus. The nucleus globosus is an elongated mass directed anterior-posteriorly and placed medial to the preceding. The nucleus fastigii is somewhat larger than the other 2, and is situated close to the middle line at the anterior end of the superior vermis, and immediately over the roof of the fourth ventricle, from which it is separated by a thin layer of white substance. The cerebellum is concerned with the coordination of movements necessary in equilibration, locomotion, and prehension. In it terminate pathways conducting impulses of muscle sense, tendon sense, joint sense, and equilibratory disturbances. With the exception of the ventral spinocerebellar fasciculus, these impulses enter through the inferior peduncle. The reflex arc is completed by fibers in the superior peduncle, which pass to the red nucleus and the thalamus, and thence, by additional neurons, rubrospinal tract, to the motor centers. The exact function of its different parts are still quite uncertain, owing to the contradictory nature of the evidence furnished by 1. Ablation experiments upon animals, and 2. Clinical observation in man of the effects produced by abscesses or tumors affecting different portions of the organ. The fourth ventricle, ventriculus cortis. The fourth ventricle, or cavity of the hindbrain, is situated in front of the cerebellum and behind the pons and upper half of the medulla oblongata. Developmentally considered, the fourth ventricle consists of 3 parts: a superior belonging to the isthmus rhombencephalon, an intermediate to the metencephalon, and an inferior to the myelencephalon. It is lined by ciliated epithelium and is continuous below with the central canal of the medulla oblongata. Above, it communicates by means of a passage termed the cerebral aqueduct with the cavity of the third ventricle. It presents 4 angles and possesses a roof or dorsal wall, a floor or ventral wall, and lateral boundary. Angles. The superior angle is on a level with the upper border of the pons and is continuous with the lower end of the cerebral aqueduct. The inferior angle is on a level with the lower end of the olive and opens into the central canal of the medulla oblongata. Each lateral angle corresponds with the point of meeting of the brachia and inferior peduncle. A little below the lateral angles, on a level with the striae medullares, the ventricular cavity is prolonged outward in the form of 2 narrow lateral recesses, one on either side. These are situated between the inferior peduncles and the flocculi, and reach as far as the attachments of the glossopharyngeal and vagus nerves. Lateral boundaries. The lower part of each lateral boundary is constituted by the clava, the fasciculus cuneatus, and the inferior peduncle, the upper part by the middle and the superior peduncle. Roof or dorsal wall. The upper portion of the roof is formed by the superior peduncle and the anterior medullary velum, the lower portion by the posterior medullary velum, the epithelial lining of the ventricle covered by the telechoroidea inferior, the tanae of the fourth ventricle, and the obex. The superior peduncle, on emerging from the central white substance of the cerebellum, pass upward and forward, forming at first the lateral boundaries of the upper part of the cavity. On approaching the inferior colliculi, they converge and their medial portions overlap the cavity and form part of its roof. The anterior medullary velum fills the angular interval between the superior peduncle and is continuous behind with the central white substance of the cerebellum. It is covered on its dorsal surface by the lingula of the superior vermis. The posterior medullary velum is continued downward and forward from the central white substance of the cerebellum in front of the nodule and tonsils, and ends inferiorly in a thin, concave, somewhat ragged margin. Below this margin, the roof is devoid of nervous matter, except in the immediate vicinity of the lower lateral boundaries of the ventricle, where 2 narrow white bands, the tinae of the fourth ventricle, ligulae, appear. These bands meet over the inferior angle of the ventricle in a thin triangular lamina, the obex. The non-nervous part of the roof is formed by the epithelial lining of the ventricle, which is prolonged downward as a thin membrane from the deep surface of the posterior medullary velum to the corresponding surface of the obex and tinae, and thence on to the floor of the ventricular cavity. It is covered and strengthened by a portion of the pia mater, which is named the telechoroidea of the fourth ventricle. The tinae of the fourth ventricle, tinae ventriculi corto, ligula, are 2 narrow bands of white matter, one on either side, which complete the lower part of the roof of the cavity. Each consists of a vertical and a horizontal part. The vertical part is continuous below the obex with the clava, to which it is adherent by a lateral border. The horizontal portion extends transversely across the inferior peduncle below the striae medullares and roofs in the lower and posterior part of the lateral recess. It is attached by its lower margin to the inferior peduncle and partly encloses the choroid plexus, which, however, projects beyond it like a cluster of grapes, and hence this part of the tinae has been termed the cornucopia, Bochtelik. The obex is a thin triangular gray lamina which roofs in the lower angle of the ventricle and is attached by its lateral margins to the clavae. The telechoroidea of the fourth ventricle is the name applied to the triangular fold of pia mater, which is carried upward between the cerebellum and the medulla oblongata. It consists of 2 layers which are continuous with each other in front and are more or less adherent throughout. The posterior layer covers the anterior-inferior surface of the cerebellum, while the anterior is applied to the structures which form the lower part of the roof of the ventricle, and is continued inferiorly with the pia mater on the inferior peduncles and closed part of the medulla. Choroid plexuses. These consist of two highly vascular inflections of the tila-choroidea, which invaginate the lower part of the roof of the ventricle and are everywhere covered by the epithelial lining of the cavity. Each consists of a vertical and a horizontal portion. The former lies close to the middle line, and the latter passes into the lateral recess and projects beyond its apex. The vertical parts of the plexuses are distinct from each other, but the horizontal portions are joined in the middle line, and hence the entire structure presents the form of the letter T, the vertical limb of which, however, is double. Openings in the roof. In the roof of the fourth ventricle, there are three openings, a medial and two lateral. The medial aperture, foramen majorandi, is situated immediately above the inferior angle of the ventricle. The lateral apertures, foramina ovlushka, are found at the extremities of the lateral recesses. By means of these three openings, the ventricle communicates with the subarachnoid cavity, and the cerebral spinal fluid can circulate from the one to the other. Rhomboid fossa. Fossa rhomboidea, floor of the fourth ventricle. The anterior part of the fourth ventricle is named from its shape, the rhomboid fossa, and its anterior wall, formed by the back of the pons and medulla oblongata, constitutes the floor of the fourth ventricle. It is covered by a thin layer of gray substance continuous with that of the medulla spinalis. Superficial to this is a thin lamina of neuroglia which constitutes the ependema of the ventricle and supports a layer of ciliated epithelium. The fossa consists of three parts, superior, intermediate, and inferior. The superior part is triangular in shape and limited laterally by the superior cerebellar peduncle. Its apex, directed upward, is continuous with the cerebral aqueduct. Its base is represented by an imaginary line at the level of the upper ends of the superior foveae. The intermediate part extends from this level to that of the horizontal portions of the tinea of the ventricle. It is narrow above, where it is limited laterally by the medial peduncle, but widens below and is prolonged into the lateral recesses of the ventricle. The inferior part is triangular, and its downwardly directed apex, named the calamus scriptorius, is continuous with the central canal of the closed port of the medulla oblongata. The rhomboid fossa is divided into symmetrical halves by a median sulcus, which reaches from the upper to the lower angles of the fossa and is deeper below than above. On either side of this sulcus is an elevation, the medial eminence, bounded laterally by a sulcus, the sulcus limitans. In the superior part of the fossa, the medial eminence has a width equal to that of the corresponding half of the fossa, but opposite the superior fovea, it forms an elongated swelling, the colliculus facialis, which overlies the nucleus of the abducent nerve, and is, in part at least, produced by the ascending portion of the root of the facial nerve. In the inferior part of the fossa, the medial eminence assumes the form of a triangular area, the trigonum hypoglossae. When examined under water with a lens, this trigone is seen to consist of a medial and a lateral area separated by a series of oblique furrows. The medial area corresponds with the upper part of the nucleus of the hypoglossal nerve, the lateral, with a small nucleus, the nucleus intercalatus. The sulcus limitans forms the lateral boundary of the medial eminence. In the superior part of the rhomboid fossa, it corresponds with the lateral limit of the fossa and presents a bluish-gray area, the locus ceruleus, which owes its color to an underlying patch of deeply pigmented nerve cells termed the substantia ferruginea. At the level of the colliculus facialis, the sulcus limitans widens into a flattened depression, the superior fovea, and in the inferior part of the fossa appears as a distinct dimple, the inferior fovea. Lateral to the foveae is a rounded elevation named the area acoustica, which extends into the lateral recess and there forms a feebly marked swelling, the tuberculum acousticum. Winding around the inferior peduncle and crossing the area acoustica and the medial eminence are a number of white strands, the striae medullares, which form a portion of the cochlear division of the acoustic nerve and disappear into the median sulcus. Below the inferior fovea and between the trigonum hypoglossae and the lower part of the area acoustica is a triangular dark field, the alis cinerea, which corresponds to the sensory nucleus of the vagus and glossopharyngeal nerves. The lower end of the ala cinerea is crossed by a narrow translucent ridge, the funiculus separans, and between this funiculus and the clava is a small tongue-shaped area, the area postrema. On section, it is seen that the funiculus separans is formed by a strip of thickened ependema, and the area postrema by loose, highly vascular neuroglial tissue containing nerve cells of moderate size. End of section 11. Section 12 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Midbrain. The midbrain or mesencephalon. The midbrain or mesencephalon is the short constricted portion which connects the pons and cerebellum with the thalamoencephalon and cerebral hemispheres. It is directed upward and forward and consists of 1. A ventrolateral portion composed of a pair of cylindrical bodies named as cerebral peduncles. 2. A dorsal portion consisting of 4 rounded eminences named the corpora quadrigemina. And 3. An intervening passage or tunnel, the cerebral aqueduct, which represents the original cavity of the midbrain and connects the 3rd with the 4th ventricles. The cerebral peduncles, pedunculus cerebri, crus cerebri, are 2 cylindrical masses situated at the base of the brain and largely hidden by the temporal lobes of the cerebrum, which must be drawn aside or removed in order to expose them. They emerge from the upper surface of the pons, one on either side of the middle line, and, divergent as they pass upward and forward, disappear into the substance of the cerebral hemispheres. The depressed area between the crura is termed the interpeduncular fossa and consists of a layer of grayish substance, the posterior perforated substance, which is pierced by small apertures for the transmission of blood vessels. Its lower part lies on the ventral aspect of the medial portions of the tegmenta and contains a nucleus named the interpeduncular ganglion. Its upper part assists in forming the floor of the 3rd ventricle. The ventral surface of each peduncle is crossed from the medial to the lateral side by the superior cerebellar and posterior cerebral arteries. Its lateral surface is in relation to the gyrus hippocampi of the cerebral hemisphere and is crossed from behind forward by the trochlear nerve. Close to the point of disappearance of the peduncle into the cerebral hemisphere, the optic tract winds forward around its ventrolateral surface. The medial surface of the peduncle forms the lateral boundary of the interpeduncular fossa and is marked by a longitudinal furrow, the oculomotor sulcus, from which the roots of the oculomotor nerve emerge. On the lateral surface of each peduncle, there is a second longitudinal furrow, termed a lateral sulcus. The fibers of the lateral lemniscus come to the surface in this sulcus and pass backward and upward to disappear under the inferior colliculus. Structure of the cerebral peduncles. On transverse section, each peduncle is seen to consist of a dorsal and a ventral part, separated by a deeply pigmented lamina of gray substance termed the substantia nigra. The dorsal part is named the tegmentum, the ventral the base or crusta. The 2 bases are separated from each other, but the tegmenta are joined in the median plane by a forward prolongation of the raphe of the pons. Laterally, the tegmenta are free. Dorsally, they blend with the corpora quadrigemina. The base, basis pedunculi, crusta or pes, is semilunar on transverse section and consists almost entirely of longitudinal bundles of efferent fibers, which arise from the cells of the cerebral cortex and are grouped into 3 principal sets, that is, cerebrospinal, frontopontine, and temporopontine. The cerebrospinal fibers, derived from the cells of the motor area of the cerebral cortex, occupy the middle 3/5 of the base. They are continued partly to the nuclei of the motor cranial nerves, but mainly into the pyramids of the medulla oblongata. The frontopontine fibers are situated in the medial 1/5 of the base. They arise from the cells of the frontal lobe and end in the nuclei of the pons. The temporopontine fibers are lateral to the cerebrospinal fibers. They originate in the temporal lobe and end in the nuclei pontus. The substantia nigra, intercalatum, is a layer of gray substance containing numerous deeply pigmented multipolar nerve cells. It is semilunar on transverse section, its concavity being directed towards the tegmentum. From its convexity, prolongations extend between the fibers of the base of the peduncle. Thicker medially than laterally, it reaches from the oculomotor sulcus to the lateral sulcus, and extends from the upper surface of the pons to the subthalamic region. Its medial part is traversed by the fibers of the oculomotor nerve as these stream forward to reach the oculomotor sulcus. The connections of the cells of the substantia nigra have not been definitively established. It receives collaterals from the medial lemniscus and the pyramidal bundles. Bechtereau is of the opinion that the fibers from the motor area of the cerebral cortex form synapses with the cells whose axons pass to the motor nucleus of the trigeminal nerve and serve for the coordination of the muscles of mastication. The tegmentum is continuous below with the reticular formation of the pons and, like it, consists of longitudinal and transverse fibers together with a considerable amount of gray substance. The principal gray masses of the tegmentum are the red nucleus and the interpeduncular ganglion. Of its fibers, the chief longitudinal tracts are the superior peduncle, the medial longitudinal fasciculus, and the lemniscus. Gray substance. The red nucleus is situated in the anterior part of the tegmentum and is continued upward into the posterior part of the subthalamic regions. In sections at the level of the superior colliculus, it appears as a circular mass which is traversed by the fibers of the oculomotor nerve. It receives many terminals and collaterals from the superior cerebellar peduncle, also collaterals from the ventral longitudinal bundle, from Gudin's bundle, and the median lemniscus. The axons of its larger cells cross the middle line and are continued downward into the lateral funiculus of the medulla spinalis as the rubrospinal tract, those of its smaller cells and mainly in the thalamus. The rubrospinal tract forms an important part of the pathway from the cerebellum to the lower motor centers. The interpeduncular ganglion is a median collection of nerve cells situated in the ventral part of the tegmentum. The fibers of the fasciculus retroflexus of Meinert, which have their origin in the cells of the ganglion habenula, end in it. Besides the two nuclei I mentioned, there are small collections of cells which form the dorsal and ventral nuclei, and the central nucleus or nucleus of the raphe. White substance. 1. The origin and course of the superior peduncle have already been described. 2. The medial posterior longitudinal fasciculus is continuous below with the proper fasciculi of the anterior and lateral funiculi of the medulla spinalis. In the medulla oblongata and pons, it runs close to the middle line near the floor of the fourth ventricle. In the midbrain, it is situated on the ventral aspect of the cerebral aqueduct, below the nuclei of the oculomotor and trochlear nerves. Its connections are imperfectly known, but it consists largely of ascending and descending intersegmental or association fibers, which connect in the nuclei of the hindbrain and midbrain to each other. Many of the fibers arise in Dieter's nucleus, lateral vestibular nucleus, and divide into ascending and descending branches which send terminals and collaterals to the motor nuclei of the cranial and spinal nerves. Its spinal portion is located in the anterior funiculus and is known as the vestibulospinal fasciculus. Other fibers pass to the median longitudinal bundle from cells in the reticular formation of the medulla, pons, and midbrain, and also from certain large cells in the terminal nucleus of the trigeminal nerve. According to Edinger, it extends to the so-called nucleus of the posterior longitudinal bundle in the hypothalamic region. But this is uncertain, and the fibers above the nucleus of the oculomotor are smaller in diameter than the rest of the bundle. According to Held, fibers from the posterior commissure can be traced into the posterior longitudinal bundle, and according to the same author, many of the descending fibers arise in the superior colliculus, and, after decussating in the middle line, end in the motor nuclei of the pons and medulla oblongata. These fibers from the superior colliculus probably pass into the ventral longitudinal bundle. Fibers are said to pass through the medial longitudinal fasciculus from the nucleus of the abducent into the oculomotor nerve of the opposite side, and through this nerve to the rectus medialis oculi. Fraser, however, denies the existence of such fibers. Again, fibers are said to be prolonged through this fasciculus from the nucleus of the oculomotor nerve into the facial nerve and are distributed to the orbicularis oculi, the corrugator, and the frontalis. The ventral to longitudinal bundle consists for the most part of the tectospinal fasciculus and arises from the superior colliculus. The fibers arch ventrally around the central gray matter and cross the midline in the fountain decussation of Monert. They then descend in the tegmentum, part of them passing through the red nucleus ventral to the medial longitudinal bundle. In the medulla oblongata and spinal cord, its fibers are more or less intermingled with the medial longitudinal bundle and the rubrospinal tract. It descends in the adjoining region of the ventral and lateral funiculi. Collaterals and terminals are given off to the red nucleus and probably other nuclei of the brainstem and to the anterior column of the spinal cord. It is probably concerned in optic reflexes. The medial lemniscus or medial fillet. The fibers of the medial lemniscus take origin in the gracile and cuneate nuclei of the medulla oblongata, and as internal arcuate fibers, they cross to the opposite side in the sensory decussation. They then pass into interolivary stratum upward through the medulla oblongata, in which they are situated behind the cerebrospinal fibers and between the olives. In the pons and lower part of the midbrain, it occupies the ventral part of the reticular formation and tegmentum close to the raphe, while above it gradually shifts to the dorsolateral part of the tegmentum in the angle between the red nucleus and the substantia nigra. In the pons, it assumes a flattened ribbon-like appearance and is placed dorsal to the trapezium. As the lemniscus ascends, it receives additional fibers from the terminal sensory nuclei of the cranial nerves of the opposite side. Many of the fibers which arise from the terminal sensory nuclei of the cranial nerves pass upward in the formatio reticularis as a separate bundle, known as the central tract of the cranial nerves, to the thalamus. Many fibers either terminate in or send off collaterals to the gray matter of the medulla, the pons, and the midbrain. Large numbers of fibers pass to or from the substantia nigra. Many collaterals enter the red nucleus, and other fibers are said to run to the superior colliculus. The great bulk of the fibers, however, enter the ventrolateral portion of the thalamus, give off collaterals to the posterior semilunar nucleus, and then terminate in the principal central nucleus of the thalamus. In the cerebral peduncle, a few of its fibers pass upward in the lateral part of the base of the peduncle on the dorsal aspect of the temporopontine fibers, and reach the lentiform nucleus and the insula. The greater part of the medial lemniscus, on the other hand, is prolonged through the tegmentum, and most of its fibers end in the thalamus. Probably some are continued directly through the occipital part of the internal capsule to the cerebral cortex. From the cells of the thalamus, a relay of fibers is prolonged to the cerebral cortex. The medial lemniscus may be considered as the upward continuation of the posterior funiculus of the spinal cord and to convey conscious impulses of muscle sense and tactile discrimination. The central or thalamic tract of the cranial nerves is closely associated with the medial lemniscus. The fibers of the spinothalamic fasciculi are continued from the spinal cord into this tract, which passes upward in the reticular formation and the tegmentum to the thalamus, along the dorsal side of the median lemniscus. It receives fibers from the opposite terminal sensory nuclei of the vagus, glossopharyngeal, facial, trigeminal, and probably the vestibular nerves. Many of the secondary sensory fibers of the trigeminal cross the raphe from its terminal nucleus and pass upward to the thalamus by a more or less separate but closely associated pathway known as the central tract of the trigeminal nerve, which also lies on the dorsal aspect of the lemniscus. These two tracts give off collaterals to the posterior semilunar nucleus of the thalamus and terminate in the anterior semilunar nucleus of the ventrolateral region of the thalamus, sending collaterals into the zona incerta. The fibers of the rubrospinal tract, bundle of Monaco, arise in the red nucleus, cross the midline in the decussation of Forel, and pass downward in the formatio reticularis of the brainstem to the lateral funiculus of the spinal cord, ventral to the crossed pyramidal tract. The lateral lemniscus, lemniscus lateralis, comes to the surface of the midbrain along its lateral sulcus and disappears under the inferior colliculus. It consists of fibers from the terminal nuclei of the cochlear division of the acoustic nerve, together with others from the superior olivary and trapezoid nuclei. Most of these fibers are crossed, but some are uncrossed. Many of them pass to the inferior colliculus of the same or opposite side, but others are prolonged to the thalamus and thence through the occipital part of the internal capsule to the middle and superior temporal gyri. The corpora quadrigemina are four rounded eminences which form the dorsal part of the midbrain. They are situated above and in front of the anterior medullary velum and superior peduncle, and below and behind the third ventricle and posterior commissure. They are covered by the splenium of the corpus callosum and are partly overlapped on either side by the medial angle or pulvinar of the posterior end of the thalamus. On the lateral aspect, under cover of the pulvinar, is an oval eminence named the medial geniculate body. The corpora quadrigemina are arranged in pairs, superior and inferior colliculi, and are separated from one another by a crucial sinus. The longitudinal part of this sulcus expands superiorly to form a slight depression which supports the pineal body, a cone-like structure which projects backwards from the thalamoencephalon and partly obscures the superior colliculi. From the inferior end of the longitudinal sulcus, a white band, termed the frenulum veli, is prolonged downward to the anterior medullary velum. On either side of this band, the trochlear nerve emerges and passes forward on the lateral aspect of the cerebral peduncle to reach the base of the brain. The superior colliculi are larger and darker in color than the inferior, and are oval in shape. The inferior colliculi are hemispherical and somewhat more prominent than the superior. The superior colliculi are associated with the sense of sight, the inferior with that of hearing. From the lateral aspect of each colliculus, a white band, termed the brachium, is prolonged upward and forward. The superior brachium extends lateralward from the superior colliculus, and, passing between the pulvinar and the medial geniculate body, is partly continued into an eminence called the lateral geniculate body, and partly into the optic tract. The inferior brachium passes forward and upward from the inferior colliculus and disappears under cover of the medial geniculate body. In close relationship with the corpora quadrigemina are the superior peduncles, which emerge from the upper and medial parts of the cerebellar hemispheres. They run upward and forward and, passing under the inferior colliculi, enter the tegmenta as already described. Structure of the corpora quadrigemina. The inferior colliculus, colliculus inferior, inferior quadrigeminal body, postgemina, consists of a compact nucleus of gray substance containing large and small multipolar nerve cells, and more or less completely surrounded by white fibers derived from the lateral lemniscus. Most of these fibers end in the gray nucleus of the same side, but some cross the middle line and end in that of the opposite side. From the cells of the gray nucleus, fibers are prolonged through the inferior brachium into the tegmentum of the cerebral peduncle, and are carried to the thalamus and the cortex of the temporal lobe. Other fibers cross the middle line and end in the opposite colliculus. The superior colliculus, colliculus superior, superior quadrigeminal body, pregemina, is covered by a thin stratum, stratum zonale, of white fibers, the majority of which are derived from the optic tract. Beneath this is the stratum cinereum, a cap-like layer of gray substance, thicker in the center than at the circumference, and consisting of numerous small multipolar nerve cells embedded in a fine network of nerve fibers. Still deeper is the stratum opticum, containing large multipolar nerve cells separated by numerous fine nerve fibers. Finally, there is the stratum lemnisci, consisting of fibers derived partly from the lemniscus and partly from the cells of the stratum opticum. Interspersed among these fibers are many large multipolar nerve cells. The two last named strata are sometimes termed the gray-white layers, from the fact they consist of both gray and white substance. Of the afferent fibers which reach the superior colliculus, some are derived from the lemniscus, but the majority have their origins in the retina and are conveyed to it through the superior brachium. All of them end by arborizing around the cells of the gray substance. Of the efferent fibers, some cross the middle line to the opposite colliculus, many ascend through the superior brachium, and finally reach the cortex of the occipital lobe of the cerebrum, while others, after undergoing decussation, fountain decussation of Meinert, form the tectospinal fasciculus, which descends through the formatio reticularis of the midbrain, pons, and medulla oblongata into the medulla spinalis, where it is found partly in the anterior funiculus and partly intermingled with the fibers of the rubrospinal tract. The corpora quadrigemina are larger in the lower animals than in man. In fishes, reptiles, and birds they are hollow, and only 2 in number. Corpora bigemina. They represent the superior colliculi of mammals and are frequently termed the optic lobes because of their intimate connection with the optic tracts. The cerebral aqueduct, Aqueductus cerebri, aqueductus sylvius, is a narrow canal about 15 millimeters long, situated between the corpora quadrigemina and tegmenta, and connecting the third with the fourth ventricle. Its shape, as seen in transverse section, varies at different levels, being T-shaped, triangular above, and oval in the middle. The central part is slightly dilated, and was named by Retzius the ventricle of the midbrain. It is lined by ciliated columnar epithelium, and is surrounded by a layer of gray substance named the central gray stratum. This is continuous below with the gray substance in the rhomboid fossa, and above with that of the third ventricle. Dorsally, it is partly separated from the gray substance of the quadrigeminal bodies by the fibers of the lemniscus. Ventral to it are the medial longitudinal fasciculus and the formatio reticularis of the tegmentum. Scattered throughout the central gray stratum are numerous nerve cells of various sizes, interlaced by a network of fine fibers. Besides these scattered cells, it contains three groups which constitute the nuclei of the oculomotor and trochlear nerves and the nucleus of the mesencephalic root of the trigeminal nerve. The nucleus of the trigeminal nerve extends along the entire length of the aqueduct and occupies the lateral part of the gray stratum, while the nuclei of the oculomotor and trochlear nerves are situated in its ventral part. The nucleus of the oculomotor nerve is about 10 millimeters long and lies under the superior colliculus, beyond which, however, it extends for a short distance into the gray substance of the third ventricle. The nucleus of the trochlear nerve is small and nearly circular, and is on a level with the plane carried transversely through the upper part of the inferior colliculus. End of section 12. Section 13 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information to the volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 1. The forebrain or prosencephalon. The forebrain or prosencephalon consists of 1. The diencephalon, corresponding in a large measure to the third ventricle and the structures which bound it, and 2. The telencephalon, comprising the largest part of the brain, that is, the cerebral hemispheres. These hemispheres are intimately connected with each other across the middle line, and each contains a large cavity named the lateral ventricle. The lateral ventricles communicate through the intervertebral foramen with the third ventricle, but are separated from each other by a medial septum, the septum pellucidum. This contains a slit-like cavity which does not communicate with the ventricles. The diencephalon. The diencephalon is connected above and in front with the cerebral hemispheres, behind with the midbrain. Its upper surface is concealed by the corpus callosum and is covered by a fold of pia mater named the telechoroidea of the third ventricle. Inferiorly, it reaches to the base of the brain. The diencephalon comprises 1. The thalamencephalon, 2. The pars mammillaris hypothalami, and 3. The posterior part of the third ventricle. For descriptive purposes, however, it is more convenient to consider the whole of the third ventricle and its boundaries together. This necessitates the inclusion under this heading of the pars optica hypothalami and the corresponding part of the third ventricle, structures which properly belong to the telencephalon. The thalamencephalon. The thalamencephalon comprises 1. The thalamus, 2. The metathalamus or corpora geniculata, and 3. The epithalamus, consisting of the trigone habenulae, the pineal body, and the posterior commissure. The thalami, optic thalamus, are 2 large ovoid masses situated one on either side of the third ventricle, and reaching for some distance behind that cavity. Each measures about 4 centimeters in length, and presents 2 extremities, an anterior and a posterior, and 4 surfaces, superior, inferior, medial, and lateral. The anterior extremity is narrow. It lies close to the middle line and forms the posterior boundary of the interventricular foramen. The posterior extremity is expanded, directed backward and lateralward, and overlaps the superior colliculus. Medially, It presents an angular prominence, the pulvinar, which is continued laterally into an oval swelling, the lateral geniculate body, while beneath the pulvinar, but separated from it by the superior brachium, is a second oval swelling, the medial geniculate body. The superior surface is free, slightly convex, and covered by a layer of white substance, termed a stratum zonale. It is separated laterally from the caudate nucleus by a white band, the stria terminalis, and by the terminal vein. It is divided into a medial and a lateral portion by an oblique shallow furrow, which runs from behind forward and medialward and corresponds with the lateral margin of the fornix. The lateral part forms a portion of the floor of the lateral ventricle and is covered by the epithelial lining of this cavity. The medial part is covered by the telechoroidea of the third ventricle and is destitute of an epithelial covering. In front, the superior is separated from the medial surface by a salient margin, the tinea thalami, along which the epithelial lining of the third ventricle is reflected onto the undersurface of the telechoroidea. Behind, it is limited medially by a groove, the sulcus habenulae, which intervenes between it and a small triangular area termed the trigrome habenulae. The inferior surface rests upon and is continuous with the upward prolongation of the tegmentum, subthalamic tegmental region, in front of which it is related to substantia innominata of Meinert. The medial surface constitutes the upper part of the lateral wall of the third ventricle and is connected to the corresponding surface of the opposite thalamus by a flattened gray band, the massa intermedia, middle or gray commissure. This mass averages about 1 centimeter in its anterior-posterior diameter. It sometimes consists of 2 parts and occasionally is absent. It contains nerve bundles and nerve fibers. A few of the latter may cross the middle line, but most of them pass towards the middle line and then curve lateralward on the same side. The lateral surface is in contact with a thick band of white substance which forms the occipital part of the internal capsule and separates the thalamus from the lentiform nucleus of the corpus striatum. Structure. The thalamus consists chiefly of gray substance, but its upper surface is covered by a layer of white substance termed a stratum zonale, and its lateral surface by a similar layer termed the lateromedullary lamina. Its gray substance is incompletely subdivided into 3 parts, anterior, medial, and lateral, by a white layer, the medial medullary lamina. The anterior part comprises the anterior tubercle, the medial part lies next to the lateral wall of the third ventricle, while the lateral and largest part is interposed between the medullary laminae and includes the pulvinar. The lateral part is traversed by numerous fibers which radiate from the thalamus into the internal capsule and pass through the latter into the cerebral cortex. These 3 parts are built up of numerous nuclei, the connections of many of which are imperfectly known. Connections. The thalamus may be regarded as a large ganglionic mass in which the ascending tracts of the tegmentum and a considerable portion of the fibers of the optic tract end, and from the cells in which numerous fibers, thalamocortical, take origin and radiate to almost every part of the cerebral cortex. The lemniscus, together with other longitudinal strands of the tegmentum, enters its ventral part, the thalamo-mammary fasciculus, bundle of Vic d'Azur, from the corpus mammillare, enters in its anterior tubercle, while many of its fibers of the optic tract terminate in its posterior ends. The thalamus also receives numerous fibers, corticothalamic, from the cells of the cerebral cortex. The fibers that arise from the cells of the thalamus form 4 principal groups or stalks. A. Those of the anterior stalk pass through the frontal part of the internal capsule to the frontal lobe. B. The fibers of the posterior stalk, optic radiations, arise in the pulvinar and are conveyed through the occipital part of the internal capsule to the occipital lobe. C. The fibers of the inferior stalk leave the undermedial surface of the thalamus and pass beneath the lentiform nucleus to the temporal lobe and insula. D. Those of the parietal stalk pass from the lateral nucleus of the thalamus to the parietal lobe. Fibers also extend from the thalamus into the corpus striatum. Those destined for the caudate nucleus leave the lateral surface, and those from the lentiform nucleus, the inferior surface of the thalamus. The metathalamus comprises the geniculate bodies, which are 2 in number, a medial and a lateral on each side. The medial geniculate body, corpus geniculatum mediale, internal geniculate body, post geniculatum, lies under cover of the pulvinar of the thalamus and on the lateral aspect of the corpora quadrigeminum. Oval in shape, with its long axis directed forward and lateralward, it is lighter in color and smaller in size than the lateral. The inferior brachium from the inferior colliculus disappears under cover of it, while from its lateral extremity, a strand of fibers passes to join the optic tract. Entering it are many acoustic fibers from the lateral lemniscus. The medial geniculate bodies are connected with one another by the commissure of Gudin, which passes through the posterior part of the optic chiasma. The lateral geniculate body, corpus geniculatum laterale, external geniculate body, pregeniculatum, is an oval elevation on the lateral part of the posterior end of the thalamus, and is connected with the superior colliculus by the superior brachium. It is of a dark color, and presents a laminated arrangement consisting of alternating layers of gray and white substance. It receives numerous fibers from the optic tract, while other fibers of this tract pass over or through it into the pulvinar. Its cells are large and pigmented. Their axons pass to the visual area in the occipital part of the cerebral cortex. The superior colliculus, the pulvinar, and the lateral geniculate body receive many fibers from the optic tracts, and are therefore intimately connected with sight, constituting what are termed the lower visual centers. Extirpation of the eyes in the newly born animal entails an arrest to the development of these centers, but has no effect on the medial geniculate bodies or on the inferior colliculi. Moreover, the latter are well developed in the mole, an animal in which the superior colliculi are rudimentary. The epithalamus comprises the trigonum habenulae, the pineal body, and the posterior commissure. The trigonum habenulae is a small depressed triangular area situated in front of the superior colliculus and on the lateral aspect of the posterior part of the tinea thalami. It contains a group of nerve cells termed the ganglion habenulae. Fibers enter it from the stalk of the pineal body, and others, forming what is termed the habenula commissure, pass across the middle line to the corresponding ganglion of the opposite side. Most of its fibers are, however, directed downward and form a bundle, the fasciculus retroflexus of Meinert, which passes medial to the red nucleus and, after decussating with the corresponding fasciculus of the opposite side, ends in the interpeduncular ganglion. The pineal body, corpus pineale epiphysis, is a small, conical, reddish-gray body which lies in the depression between the superior colliculi. It is placed beneath the splenia of the corpus callosum, but is separated from this by the telechoroidea of the third ventricle, the lower layer of which envelops it. It measures about 8 millimeters in length, and its base, directed forward, is attached by a stalk or peduncle of white substance. The stalk of the pineal body divides anteriorly into two laminae, a dorsal and a ventral, separated from one another by the pineal recess of the third ventricle. The ventral lamina is continuous with the posterior commissure. The dorsal lamina is continuous with the habenular commissure and divides into two strands, the medullary striae, which run forward, one on either side, along the junction of the medial and upper surface of the thalamus to blend in front with the columns of the fornix. The posterior commissure is a rounded band of white fibers crossing the middle line on the dorsal aspect of the upper end of the cerebral aqueduct. Its fibers acquire their medullary sheaths early, but their connections have not been definitively determined. Most of them have their origin in a nucleus, the nucleus of the posterior commissure, nucleus of Darkschewitz, which lies in the central gray substance of the upper end of the cerebral aqueduct, in front of the nucleus of the oculomotor nerve. Some are probably derived from the posterior part of the thalamus and from the superior colliculus, while others are believed to be continued downward into the medial longitudinal fasciculus. The hypothalamus includes the subthalamic tegmental region and the structures forming the greater part of the floor of the third ventricle, that is, the corpora mammillaria, tuber cinereum, infundibulum hypophysis, and optic chiasma. The subthalamic tegmental region consists of the upward continuation of the tegmentum. It lies on the ventrolateral aspect of the thalamus and separates it from the fibers of the internal capsule. The red nucleus and the substantia nigra are prolonged into its lower part. In front, it is continuous with the substantia nominata of Meinert, medially with the gray substance of the floor of the third ventricle. It consists from above downward of three strata. 1. Stratum dorsale, directly applied to the undersurface of the thalamus and consisting of fine longitudinal fibers. 2. Zona incerta, a continuation forward of the formatio reticularis of the tegmentum. And 3. The corpus subthalamicum, nucleus of Lewis, a brownish mass presenting a lenticular shape on transverse section, and situated on the dorsal aspect of the fibers at the base of the cerebral peduncle. It is encapsulated by a lamina of nerve fibers and contains numerous medium-sized nerve cells, the connections of which are not as yet fully determined. The corpora mammillaria, corpus albicontia, are two round white masses, each about the size of a small pea, placed side by side below the gray substance of the floor of the third ventricle, in front of the posterior perforated substance. They consist of white substance externally and gray substance internally, the cells of the latter forming two nuclei, a medial of smaller and a lateral of larger cells. The white substance is mainly formed by the fibers of the columns of the fornix, which descend to the base of the brain and end partly in the corpora mammillaria. From the cells of the gray substance of each of the mammillary bodies, two fasciculi arise. One, the thalamo-mammillary fasciculus, bundle of Vic d'Azur, passes upward into the anterior nucleus of the thalamus. The other is directed downward into the tegmentum. Afferent fibers are believed to reach the corpus mammillaria from the medial lemniscus and from the tegmentum. The tuber cinereum is a hollow eminence of gray substance situated between the corpora mammillaria behind and the optic chiasma in front. Laterally, it is continued with the anterior perforated substance, and anteriorly with a thin lamina, the lamina terminalis. From the undersurface of the tuber cinereum, a hollow conical process, the infundibulum, projects downward and forward and is attached to the posterior lobe of the hypophysis. In the lateral part of the tuber cinereum is a nucleus of nerve cells, the basal optic nucleus of Meinhardt, while close to the cavity of the third ventricle are three additional nuclei. Between the tuber cinereum and the corpora mammillaria, a small elevation with a corresponding depression in the third ventricle is sometimes seen. Retzius has named it the eminentia sacularis and regards it as a representative of the saccus vasculosus, found in this situation in some of the lower vertebrates. The hypophysis, pituitary body, is a reddish-gray, somewhat oval mass measuring about 12.5 mm in its transverse and about 8 mm in its anterior-posterior diameter. It is attached to the end of the infundibulum and is situated in the fossa hypophyseos of the sphenoidal bone, where it is retained by a circular fold of dura mater, the diaphragma sella. This fold almost completely roofs in the fossa, leaving only a small central aperture through which the infundibulum passes. Optic chiasma. Chiasma opticum, optic commissure. The optic chiasma is a flattened and somewhat quadrilateral band of fibers situated at the junction of the floor and anterior wall of the third ventricle. Most of its fibers have their origins in the retina and reach the chiasma through the optic nerves, which are continuous with its anterior lateral angles. In the chiasma, they undergo a partial decussation. The fibers from the nasal half of the retina decussate and enter the optic tract of the opposite side, while the fibers from the temporal half of the retina do not undergo decussation, but pass back into the optic tract of the same side. Occupying the posterior part of the commissure, however, is a strand of fibers, the commissure of Guden, which is not derived from the optic nerves. It forms a connecting link between the medial geniculate bodies. Optic tracts. The optic tracts are continued backward and lateralward from the posterior lateral angles of the optic chiasma. Each passes between the anterior perforated substance and the tuber cinereum, and, winding around the ventrolateral aspect of the cerebral peduncle, divides into a medial and a lateral root. The former comprises the fibers of Guden's commissure. The lateral root consists mainly of afferent fibers which arise in the retina and undergo partial decussation in the optic chiasma as described. But it also contains a few fine efferent fibers which have their origin in the brain and their terminations in the retina. When traced backward, the afferent fibers of the lateral root are found to end in the lateral geniculate body and pulvinar of the thalamus, and in the superior colliculus, and these three structures constitute the lower visual centers. Fibers arise from the nerve cells in these centers, and pass through the occipital part of the internal capsule under the name of the optic radiations, to the cortex of the occipital lobe of the cerebrum, where the higher or cortical visual center is situated. Some of the fibers of the optic radiations take an opposite course, arising from the cells of the occipital cortex and passing to the lower visual centers. Some fibers are detached from the optic tract and pass through the cerebral peduncle to the nucleus of the ocular motor nerve. These may be regarded as the afferent branches for the sphincter pupillae and ciliaris muscles. Other fibers have been described as reaching the cerebrum through the superior peduncle, while others again are lost in the pons. The third ventricle, ventriculus tertius. The third ventricle is a median cleft between the two thalami. Behind it, it communicates with the fourth ventricle through the cerebral aqueduct, and in front with the lateral ventricles through the interventricular foramen. Somewhat triangular in shape, with the apex directed backwards, it has a roof, a floor, an anterior and a posterior boundary, and a pair of lateral walls. The roof is formed by a layer of epithelium which stretches between the upper edges of the lateral wall of the cavity and is continuous with the epithelial lining of the ventricle. It is covered by and adherent to a fold of pia mater, named the telechoroidea of the third ventricle, from the undersurface of which a pair of vascular fringe processes, the choroid plexuses of the third ventricle, project downward, one on either side of the middle line, and invaginate the epithelial roof into the ventricular cavity. The floor slopes downward and forward and is formed mainly by the structures which constitute the hypothalamus. From before backwards, these are the optic chiasma, the tuber cinereum and infundibulum, and the corpus mammillaria. Behind the last, the floor is formed by the interpeduncular fossa and the tegmenta of the cerebral peduncles. The ventricle is prolonged downward as the funnel-shaped recess, the recessus infundibuli, into the infundibulum, and to the apex of the latter, the hypophysis, is attached. The anterior boundary is constituted below by the lamina terminalis, a thin layer of gray substance stretching from the upper surface of the optic chiasma to the rostrum of the corpus callosum, above by the columns of the fornix and the anterior commissure. At the junction of the floor and anterior wall, immediately above the optic chiasma, the ventricle presents a small angular recess or diverticulum, the optic recess. Between the columns of the fornix and just above the anterior commissure is a second recess termed the vulva. At the junction of the roof and anterior wall of the ventricle, and situated between the thalami behind and the columns of the fornix in front, is the interventricular foramen, foramen of Monro, through which the third communicates with the lateral ventricles. The posterior boundary is constituted by the pineal body, the posterior commissure, and the cerebral aqueduct. A small recess, the recessus pinealis, projects into the stalk of the pineal body, while in front of and above the pineal body is a second recess, the recess suprapinealis, consisting of a diverticulum of the epithelium which forms the ventricular roof. Each lateral wall consists of an upper portion formed by the medial surface of the anterior two-thirds of the thalamus, and a lower consisting of an upward continuation of gray substance of the ventricular floor. These two parts correspond to the alar and basal laminae, respectively, of the lateral wall of the forebrain vesicle, and are separated from each other by a furrow, the sulcus of Monro, which extends from the interventricular foramen to the cerebral aqueduct. The lateral wall is limited above by the taenia thalami. The columns of the fornix curve downward in front of the interventricular foramen and then run in the lateral walls of the ventricle, where, at first, they form distinct prominences, but subsequently are lost to sight. The lateral walls are joined to each other across the cavity of the ventricle by a band of gray substance, the massa intermedia. Interpeduncular fossa. This is a somewhat lozenge-shaped area of the base of the brain, limited in front by the optic chiasma, behind by the anterior superior surface of the pons, anterolaterally by the converging optic tracts, and posterolaterally by the diverging cerebral peduncles. The structures contained in it have already been described. From behind forward, they are the posterior perforated substance, corpora mammillaria, tuber cinereum, infundibulum, and hypophysis. End of section 13. Section 14 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. M.L. Cohen, Anatomy of the Human Body, Part 4 by Henry Gray. The Forebrain, Part 2. The telencephalon. The telencephalon includes 1. The cerebral hemispheres with their cavities, the lateral ventricles, and 2. The pars optica hypothalami, and the anterior portion of the third ventricle, already described under the diencephalon. As previously stated, each cerebral hemisphere may be divided into 3 fundamental parts, that is, the rhinencephalon, the corpus striatum, and the neopallium. The rhinencephalon, associated with the sense of smell, is the oldest part of the telencephalon and forms almost the whole of the hemisphere in some of the lower animals, that is, fishes, amphibians, and reptiles. In man, it is rudimentary, whereas the neopallium undergoes great development and forms the chief part of the hemisphere. The cerebral hemispheres. The cerebral hemispheres constitute the largest part of the brain and, when viewed together from above, assume the form of an ovoid mass broader behind than in front, the greatest transverse diameter corresponding with the line connecting the two parietal eminences. The hemispheres are separated medially by a deep cleft termed a longitudinal cerebral fissure, and each possesses a central cavity, the lateral ventricle. The longitudinal cerebral fissure, Fissura cerebri longitudinalis, great longitudinal fissure, contains a sickle-shaped process of dura mater, the falx cerebri. In front and behind, the fissure extends from the upper to the under surfaces of the hemispheres and completely separates them, but its middle portion separates them for only about one-half of their vertical extent, for at this part they are connected across the middle line by a great central white commissure, the corpus callosum. In a median sagittal section, the cut corpus callosum presents the appearance of a broad arched band. Its thick posterior end, termed a splenium, overlaps the midbrain but is separated from it by the telechoroidea of the third ventricle and the pineal body. Its anterior curved end, termed the genu, gradually tapers into a thinner portion, the rostrum, which is continued downward and backward in front of the anterior commissure to join the lamina terminalis. Arching backward from immediately behind the anterior commissure to the undersurface of the splenium is a second white band named the fornix. Between this and the corpus callosum are the laminae and cavity of the septum pellucidum. Surfaces of the cerebral hemispheres. Each hemisphere presents three surfaces: lateral, medial, and inferior. The lateral surface is convex in adaptation to the concavity of the corresponding half of the vault of the cranium. The medial surface is flat and vertical, and is separated from that of the opposite hemisphere by the great longitudinal fissure and the falx cerebri. The inferior surface is of an irregular form, and may be divided into three areas: anterior, middle, and posterior. The anterior area, formed by the orbital surface of the frontal lobe, is concave and rests on the roof of the orbit and nose. The middle area is convex and consists of the undersurface of the temporal lobe. It is adapted to the corresponding half of the middle cranial fossa. The posterior area is concave, directed medialward as well as downward, and is termed the tentorial surface, since it rests upon the tentorium cerebelli, which intervenes between it and the upper surface of the cerebellum. These three surfaces are separated from each other by the following borders: A. Superior medial between the lateral and medial surfaces. B. Inferior lateral between the lateral and inferior surfaces. The anterior part of this border separating the lateral from the orbital surface is known as the superciliary border. C. Medial occipital separating the medial and tentorial surfaces, and D. Medial orbital separating the orbital from the medial surface. The anterior end of the hemisphere is named the frontal pole, the posterior the occipital pole, and the anterior end of the temporal lobe the temporal pole. About 5 centimeters in front of the occipital pole on the inferior lateral border is an indentation or notch named the preoccipital notch. The surfaces of the hemispheres are molded into a number of irregular eminences named gyri or convolutions, and separated by furrows termed fissures and sulci. The furrows are of two kinds, complete and incomplete. The former appear earlier in fetal life, are few in number, and are produced by infoldings of the entire thickness of the brain wall, and give rise to the corresponding elevations in the interior of the ventricle. They comprise the hippocampal fissure and parts of the calcarine and collateral fissures. The incomplete furrows are very numerous and only indent the subjacent white substance without producing any corresponding elevations in the ventricular cavity. The gyri and their intervening fissures and the sulci are fairly constant in their arrangement. At the same time, they vary within certain limits, not only in different individuals, but on the two hemispheres of the same brain. The convoluted condition of the surface permits a great expansion of the gray matter without the sacrifice of much additional space. The number and extent of the gyri, as well as the depth of the intervening furrows, appear to bear a direct relation to the intellectual powers of the individual. Certain of the fissures and sulci are utilized for the purpose of dividing the hemisphere into lobes, and are therefore termed interlobular. Included under this category are the lateral cerebral, parietal occipital, calcarine, and collateral fissures, the central and cingulate sulci, and the sulcus circularis. The lateral cerebral fissure, fissura cerebri lateralis sylvii, fissure of Sylvius, is a well-marked cleft on the inferior and lateral surfaces of the hemisphere and consists of a short stem which divides into 3 rami. The stem is situated at the base of the brain and commences in a depression at the lateral angle of the anterior perforated substance. From this point, it extends between the anterior part of the temporal lobe and the orbital surface of the frontal lobe and reaches the lateral surface of the hemisphere. Here it divides into 3 rami, an anterior horizontal, an anterior ascending, and a posterior. The anterior horizontal ramus passes forward about 2.5 centimeters into the inferior frontal gyrus, while the anterior ascending ramus extends upward into the same convolution for about an equal distance. The posterior ramus is the longest. It runs backward and slightly upward for about 7 centimeters and ends by an upward inflection in the parietal lobe. The central sulcus, sulcus centralis Rolandi, fissure of Rolando, central fissure, is situated about the middle of the lateral surface of the hemisphere and begins in or near the longitudinal cerebral fissure, a little behind its midpoint. It runs sinuously downward and forward and ends a little above the posterior ramus of the lateral fissure, and about 2.5 centimeters behind the anterior ascending ramus of the same fissure. It described 2 chief curves, a superior genu with its concavity directed forward, and an inferior genu with its concavity directed backward. The central sulcus forms an angle opening forward of about 70 degrees with the median plane. The parieto-occipital fissure, fissura parietal occipitalis, only a small part of this fissure is seen on the lateral surface of the hemisphere, its chief part being on the medial surface. The lateral part of the parieto-occipital fissure is situated about 5 centimeters in front of the occipital pole of the hemisphere and measures about 1.25 centimeters in length. The medial part of the parietal occipital fissure runs downward and forward as a deep cleft on the medial surface of the hemisphere and joins the calcarine fissure below and behind the posterior end of the corpus callosum. In most cases, it contains a submerged gyrus. The calcarine fissure, fissura calcarina, is on the medial surface of the hemisphere. It begins near the occipital pole in 2 converging rami and runs forward to a point a little below the splenium of the corpus callosum, where it is joined at an acute angle by the medial part of the parietal occipital fissure. The anterior part of this fissure gives rise to the prominence of the calcar avus and the posterior cornua of the lateral ventricle. The cingulate sulcus, sulcus cinguli, callosum marginal fissure, is on the medial surface of the hemisphere. It begins below the anterior end of the corpus callosum and runs upward and forward nearly parallel to the rostrum of this body and, curving in front of the genu, is continued backward above the corpus callosum and finally ascends to the superior medial border of the hemisphere, a short distance behind the upper end of the central sulcus. It separates the superior frontal from the cingulate gyrus. The collateral fissure, fissura collateralis, is on the tentorial surface of the hemisphere and extends from near the occipital pole to within a short distance of the temporal pole. Behind, it lies below and lateral to the calcarine fissure, from which it is separated by the lingual gyrus. In front, it is situated between the hippocampal gyrus and the anterior part of the fusiform gyrus. The sulcus circularis, circuminsular fissure, is on the lower and lateral surfaces of the hemisphere. It surrounds the insula and separates it from the frontal, parietal, and temporal lobes. The lobes of the hemispheres. By means of these fissures and sulci, assisted by certain arbitrary lines, each hemisphere is divided into the following lobes: the frontal, the parietal, the temporal, the occipital, the limbic, and the insula. Frontal lobe, lobus frontalis. On the lateral surface of the hemisphere, this lobe extends from the frontal pole to the central sulcus, the latter separating it from the parietal lobe. Below, It is limited by the posterior ramus of the lateral fissure, which intervenes between it and the central lobe. On the medial surface, it is separated from the cingulate gyrus by the cingulate sulcus, and on the inferior surface, it is bounded behind by the stem of the lateral fissure. The lateral surface of the frontal lobe is traversed by 3 sulci which divide it into 4 gyri. The sulci are named the precentral and the superior and inferior frontal. The gyri are the anterior central and the superior, middle, and inferior frontal. The precentral sulcus runs parallel to the central sulcus and is usually divided into an upper and a lower part. Between it and the central sulcus is the anterior central gyrus. From the precentral sulcus, the superior and inferior frontal sulci run forward and downward and divide the remainder of the lateral surface of the lobe into 3 parallel gyri, named respectively the superior, middle, and inferior frontal gyri. The anterior central gyrus, gyrus centralis anterior, ascending frontal convolution, precentral gyre, is bounded in front by the precentral sulcus, behind by the central sulcus. It extends from the superior medial border of the hemisphere to the posterior ramus of the lateral fissure. The superior frontal gyrus, gyrus frontalis superior, superior frontal gyrus, is situated above the superior frontal sulcus and is continued on to the medial surface of the hemisphere. The portion on the lateral surface of the hemisphere is usually more or less completely subdivided into an upper and a lower part by the anterior-posterior sulcus, the paramedian sulcus, which, however, is frequently interrupted by bridging gyri. The middle frontal gyrus, gyrus frontalis medius, medial frontal gyre, between the superior and inferior frontal sulci, is continuous with the anterior orbital gyrus on the inferior surface of the hemisphere. It is frequently subdivided into 2 by a horizontal sulcus, the medial frontal sulcus of Eberstaller, which ends anteriorly in a wide bifurcation. The inferior frontal gyrus, gyrus frontalis inferior, subfrontal gyre, lies below the inferior frontal sulcus and extends forward from the lower part of the precentral sulcus. It is continuous with the lateral and posterior orbital gyri on the undersurface of the lobe. It is subdivided by the anterior horizontal and ascending rami of the lateral fissure into 3 parts. 1. The orbital part, below the anterior horizontal ramus of the fissure. 2. The triangular part, cap of Broca, between the ascending and horizontal rami. And 3. The basilar part, behind the anterior ascending ramus. The left inferior frontal gyrus is, as a rule, more highly developed than the right, and is named the gyrus of Broca, from the fact that Broca described it as the center for articulate speech. The inferior or orbital surface of the frontal lobe is concave and rests on the orbital plate of the frontal bone. It is divided into 4 orbital gyri by a well-marked H-shaped orbital sulcus. These are named from their position the medial, anterior, lateral, and posterior orbital gyri. The medial orbital gyrus presents a well-marked anterior-posterior sulcus, the olfactory sulcus, for the olfactory tract. The portion medial to this is termed a straight gyrus and is continuous with the superior frontal gyrus on the medial surface. The medial surface of the frontal lobe is occupied by the medial part of the superior frontal gyrus, marginal gyrus. It lies between the cingulate sulcus and the superior medial margin of the hemisphere. The posterior part of this gyrus is sometimes marked off by a vertical sulcus and is distinguished as the paracentral lobule, because it is continuous with the anterior and posterior central gyri. Parietal lobe, lobus parietalis. The parietal lobe is separated from the frontal lobe by the central sulcus, but its boundaries below and behind are not so definite. Posteriorly, it is limited by the parietal occipital fissure and by a line carried across the hemisphere from the end of the fissure towards the preoccipital notch. Below, it is separated from the temporal lobe by the posterior ramus of the lateral fissure and by a line carried backward from it to meet the line passing downward to the preoccipital notch. The lateral surface of the parietal lobe is cleft by a well-marked furrow, the intraparietal sulcus of Turner, which consists of an oblique and a horizontal portion. The oblique part is named the postcentral sulcus and commences below, about midway between the lower end of the central sulcus and the upturned end of the lateral fissure. It runs upward and backward parallel to the central sulcus and is sometimes divided into an upper and a lower ramus. It forms the hinder limit of the posterior central gyrus. From about the middle of the post-central sulcus, or from the upper end of its inferior ramus, the horizontal portion of the intraparietal sulcus is carried backward and slightly upward on the parietal lobe and is prolonged under the name of the occipital ramus onto the occipital lobe, where it divides into two parts, which form nearly a right angle with the main stem and constitute the transverse occipital sulcus. The part of the parietal lobe above the horizontal portion of the intraparietal sulcus is named the superior parietal lobule, the part below, the inferior parietal lobule. The posterior central gyrus, gyrus centralis posterior, ascending parietal convolution, post-central gyre, extends from the longitudinal fissure above to the posterior ramus of the lateral fissure below. It lies parallel with the anterior central gyrus with which it is connected below and also sometimes above the central sulcus. The superior parietal lobule, lobulus parietalis superior, is bounded in front by the upper part of the post-central sulcus, but is usually connected with the posterior central gyrus above the end of the sulcus. Behind it is the lateral part of the parietal occipital fissure, around the end of which it is joined to the occipital lobe by a curved gyrus, the arcus parietal occipitalis. Below, it is separated from the inferior parietal lobule by the horizontal portion of the intraparietal sulcus. The inferior parietal lobule, lobulus parietalis inferior, subparietal district or lobule, lies below the horizontal portion of the intraparietal sulcus and behind the lower part of the post-central sulcus. It is divided from before backward into two gyri. 1. The supramarginal arches over the upturned end of the lateral fissure. It is continuous in front with the post-central gyrus and behind with the superior temporal gyrus. The second, the angular, arches over the posterior end of the superior temporal sulcus, behind which it is continuous with the middle temporal gyrus. The medial surface of the parietal lobe is bounded behind the medial part of the parietal occipital fissure. In front, by the posterior end of the cingulate sulcus, and below, it is separated from the cingulate gyrus by the subparietal sulcus. It is of small size and consists of a square-shaped convolution, which is termed the precuneus or quadrate lobe. Lobus occipitalis. The occipital lobe is small and pyramidal in shape. It presents three surfaces: lateral, medial, and tentorial. The lateral surface is limited in front by the lateral part of the parietal occipital fissure and by a line carried from the end of this fissure to the preoccipital notch. It is traversed by the transverse occipital and lateral occipital sulci. The transverse occipital sulcus is continuous with the posterior end of the occipital ramus of the intraparietal sulcus and runs across the upper part of the lobe a short distance behind the parietal occipital fissure. The lateral occipital sulcus extends from behind forward and divides the lateral surface of the occipital lobe into a superior and inferior gyrus, which are continuous in front with the parietal and temporal lobes. The medial surface of the occipital lobe is bounded in front by the medial part of the parietal occipital fissure and is traversed by the calcarine fissure, which subdivides it into the cuneus and the lingual gyrus. The cuneus is a wedge-shaped area between the calcarine fissure and the medial part of the parietal occipital fissure. The lingual gyrus lies between the calcarine fissure and the posterior part of the collateral fissure. Behind, it reaches the occipital pole. In front, it is continued on to the tentorial surface of the temporal lobe and joins the hippocampal gyrus. The tentorial surface of the occipital lobe is limited in front by an imaginary transverse line through the preoccipital notch and consists of the posterior part of the fusiform gyrus, occipitotemporal convolution, and the lower part of the lingual gyrus, which are separated from each other by the posterior segment of the collateral fissure. End of section 14.